I hope it is visible, right?
Yes, sir. Visible. Multiple sequence alignment.
Yeah, I just better close my video, otherwise it will interrupt much in between. So you can see, right? Hello.
Yes, sir.
Okay, fine.
Screen is visible.
So today's discussion will be basically on multiple sequence alignment process and what is the importance of this multiple sequence alignment and how we can do this sequence alignment, and ultimately what kind of algorithms are there when you are doing this multiple sequence alignment process. And next, our discussion will be there on which is very much connected and related with the multiple sequence alignment, that is the phylogenetic tree construction, and what is the phylogenetic tree and what are the various features are there in any phylogenetical tree, and what is the various mode and step we can follow for making phylogenetical tree, and there also what type of algorithm we are following to construct the phylogenetical tree. So that particular discussions will be in my today's lecture. So first I'll start with the multiple sequence alignment. So yesterday I had a discussion on pairwise sequence alignment. So pairwise sequence alignment, I explained the two different consequences where both the sequence of your own choice you can compare. Otherwise, your sequence you can compare with the database, any kind of sequence. So when we are comparing with the database sequence, there is having the two different method. One is the BLAST and second one is the FASTA. And if you're doing the your own choice sequence alignment, then yeah, EMBL EBI, that particular web server you are using, and you are comparing between the sequence. And of course, there is having global sequence alignment, local sequence alignment method. So all those aspects we have discussed last class, and today it will be continuation of that. That is a multiple sequence alignment. So yesterday we came to know these are the two sequence how to be aligned and what is the gap and why the gap is important and how the gap is going to create it, etc. And today I'll talk about the multiple sequence alignment. If you look at this particular picture where almost more than 10 or 11 sequences, we have put it one after another. And after putting one after another, we are trying to find out some kind of conclusions. So this method or this particular picture is indicating this is the multiple sequence alignment method. So what is how we can define the multiple sequence alignment? So here, if you are taking three sequence or more than three sequence one after another, and if you are comparing between them, the process will be considered as the multiple sequence alignment. So here the sequence could be of nucleic acid sequence, that is DNA, RNA, anything, or it could be a protein sequence. So protein sequence, you can many protein you can put one after another together, and you can take the help of various software, and you can compare between those protein, and ultimately you can get some kind of conclusions. Like that, if you're taking the nucleic acid, it could be n number of DNA, n number of RNA sequence one after another, and if you're comparing between them, that also is considering as the multiple sequence alignment. So multiple sequence alignment, why you supposed to do multiple sequence alignment? It is necessary to do to understand the what kind of domain of a particular protein, what kind of conserved sequence they are having, or if you're comparing between the DNA, what kind of particular genes having conserved that particular nucleotide compositions. To knowing that, we do the multiple sequence alignment, and major outcome of this multiple sequence alignment is to draw the phylogenetical relation between all this particular sequence. So if you wanted to create the phylogenetical tree, so first step should be there to do the multiple sequence alignment, and after making the multiple sequence alignment only you can able to reach to phylogenetical tree process. So multiple sequence alignment having huge important. So if we comparing the hemoglobin sequence whatever found in the human and hemoglobins whatever found in the primates animals, So if you compare all the hemoglobin this way by putting the multiple sequence alignment pattern, so you will able to understand which particular positions in which particular hemoglobin is having what kind of sequence, and that sequence whether it is conserved or not, that idea also you are going to get with that. And ultimately based on those hemoglobin sequence you can able to create the phylogenetical tree. So multiple sequence alignment, it is going to give a very concrete and definite idea about related sequence. Okay, it should be a related sequence when you are doing multiple sequence alignment. Suppose I have taken the hemoglobin sequence from human and I am going to compare it with the insulin of so and so and other protein of so and so. In that case, it is not going to give the a multiple alignment perfect way, and from there you may not able to create any kind of phylogenetical tree. So only one concept you need to remember, it should be all related protein or it should be all related gene sequence or mRNA sequence, then only multiple sequence alignment will be authenticated. So multiple sequence alignment, when you are doing, of course, yesterday I have explained, when you are aligning this way pairwise conditions of sequence, so there will be having certain kind of scoring pattern, and by looking that particular score we can tell this is the perfect matching or whether it is a conservative matching, what kind of matching, etc. So like that, if you're doing the multiple sequence alignment method, so there also will be having various kind of scoring function or the scoring pattern. So you need to understand that particular scoring function, how it is continuing in this particular process. And multiple sequence alignment, one of the another major important site is like in the PCR, all of you are aware that polymerase chain reactions where we can apply the specific primer and all DNTPs along with the polymerase enzyme. So there, with the help of instrument, the gene is going to amplify. So in that particular PCR, if you wanted to use a single primer and if you wanted to amplify the gene sequence of all related particular gene, so that time this multiple sequence alignment will help you a lot. So if you are able to continue the multiple sequence alignment, and from there you will get the idea which is the conserved sequence of that among present among those particular aligned sequence. So based on that, you can prepare a single primer, and a single primer, of course, it is going to amplify all kind of your related protein or related gene sequence. That is another utility of this multiple sequence alignment apart from the phylogenetical tree construction. So scoring function I was talking. So scoring function here, what we will follow, that is called the SP method, that is the summation of pairing method. So summation of pairing method, this is the example. Look at here. So suppose we have taken the three sequence and we are considering one vertical line of each. So here in this alignment, if we are taking the first line, you can see here all A, then all G, and then third is having one C, then T, then followed by A, then C, and all those stops are there. So I am taking one single line. So you need to, when the software is running and providing this SP method, so they are considering this single line from this particular alignment. So in this single line, what is happening in this example, suppose G is comparing with the T, then T is comparing with the H, and G is also going to compare with the H. And yesterday I was talking about the different matrix, all the matrix. Yesterday I was talking about the matrix. So all the matrix having a certain kind of scoring pattern. So if you are comparing with the G and T, so according to those matrix, we are going to get a particular number. Even T and S, if you are comparing, we'll be getting a number, and G and S also, if you are comparing according to the matrix, we are getting a number. So that number we are doing the summation. Each line, okay, this particular column, okay, GTS, one particular column, we are taking the summation after comparing with each alphabet with the other one. And like that, second one, third one, we are continuing. And here from this particular alignment, we are getting total score -2. Here we are getting +1. Here we are getting +6. Now, if we are taking the summation, total score is having 5. So this 5 is indicating as per the Blossom 62 matrix, this value we are getting. And here this 5 is indicating that alignment should be 2 to the power 5 mode. 2 to the power 5 means it will be 32 as per the POA algorithm, POA. So as per the POA algorithm, these 5, this whatever the summated score you are getting, that is indicating 2 to the power 5. Why 2 to the power 5? Because 2 alphabet we are considering and comparing between them, and total score is coming 5. So it will be 2 to the power 5. That means 32 times more likely to occur this particular among homologous sequences, and this particular alphabet to be present by random chance. So that means the probabilitical concept we are applying here, and this probabilitical concept is based on the POA algorithm or the POA method, POA. If you look at this particular example where 5 different sequences alignment is there, and each column is, suppose column A, column B, column C, 3 different columns is present here. So column A is having all N, column B is having 4N and 1C, column C is having 3N and 2C. Now, this particular sequence, if we are putting in this particular geometrical fashions, then we can understand that N and N will be having relations. So you can look at here, all are N present. So total possibilities and comparison should be present between N to N is a 10 number. If we go through this particular line, you will understand that total 10 number is coming. And if we come to the second line, so column B, so there how many N and N is aligning is possible, how many N and C is possible. So total it is coming, it is 6. N and this N and N, it is coming 6, whereas NC, this relation is coming 4. And in the third column, we are getting like N and N, it is 4, and N and C, between them it is the 6. So now, as per the Blossom 62 matrix, if we compare it, when N and N is matching, the score is +6, and when N and C is matching, the score is -3. So like that, the total from the first column, what is the total number we are getting based on the this 6, that means 10 into 6, 60. And the column B, we are getting for every N alignment, we are getting the 6. So 4 into 6, 24. And we are getting NC, that is also 4 number. Sorry, here 6 into 6, 36. And here we are getting NC, that is the 4. So it is each 4 alignment is getting -3. So 4, 3 are 12. So 36 minus 12, if you are doing, it is coming 24. So like that, if this particular process will be continue, and ultimately we are taking total score, that is the sum of score, and the value whatever is coming with respect to the the power we are considering, and we are thinking that is the most possible random chance of those particular sequence to be present among the homologous sequences. So in the beginning, I mentioned if you're taking the hemoglobin sequence of human and you're comparing with the XYZ protein sequence of other, it is not going to work out. It is always this homologous multiple sequence alignment. You need to continue with respect to your homologous sequence. That means the same protein or the same gene should be present among the different individual, and then among them you are going to compare those particular protein and gene sequence. So this way, basically, multiple sequence alignment pattern will work out. So now we need to understand what is the algorithm present behind this particular alignment method. So there are having basically 2 algorithmic approach. One is called exhaustive algorithmic approach, and second one, it is called basically heuristic algorithmic approach. So first I'll talk about the exhaustive algorithmic approach. So in the exhaustive algorithmic approach here, the gap penalty and the substitution matrix alignment score and the respective probability we are considering. So exactly this exhaustive algorithm will be 99% similar with the pairwise sequence alignment method. So here every 2 sequences we are taking, and between the 2 sequences and between the each alphabet of these 2 sequence we are comparing, and there we are considering the gap penalty and substitution matrix with respect to the protein and the nucleotide. So substitution matrix for the protein, it is indicating whether the similar group of amino acid is going to replace by the other, or it is going to replace by the some other group of amino acid. And in case of the nucleic acid, we are considering whether purine is going to substitute by the another purine, or pyrimidine is going to substitute by the another purine, or purine is going to substitute by the pyrimidine, or pyrimidine is going to substitute by the purine. So for nucleotide, this particular alignment for multiple sequence, it is quite easier, but with the protein, always you need to remember about the physicochemical properties of the amino acids. And based on that, we are going to create the gap penalties, everything. And here also that opening gap penalty, then extending gap penalty. So all you need to consider. And of course, this exhaustive algorithm method is going to use the dynamic matrix process. So yesterday I mentioned that why dynamic matrix is important, and based on the dynamic matrix, how that gaps are created between the two sequence. So here also dynamic matrix process only it is going to follow. So suppose A, B, C, what example previously I had given. So first A is going to compare with the C, A is going to compare with the B, and B is also going to compare with the C. So this is called the three-dimensional diagonal matrix. So these three diagonal matrix, if it is the three sequence, it is very easy to continue. But you just imagine if you are taking the 10 and 15 sequence, if you are comparing between them, then creating this way three diagonal matrix or dynamical matrix is very typical and tedious process. And there is having a chance to misread the sequences, and lot of problems may that time come, and your output whatever is going to get you are going to get that may not give the correct result one. So that means if you're using the heuristic method, that similar approach how the pairwise sequence alignment we are following, this method similar way if you are following here, that is having lot of problems. So we are trying to avoiding this particular dynamic matrix method, and we are looking for the heuristic algorithmic method. So heuristic algorithmic method is more fruitful and more accepted algorithmic process. And of course, this is related with the multiple sequence alignment method. And this heuristic algorithmic method again having three different modes. One is called progressive alignment type, second one is the iterative alignment type, and third one is the block based alignment type. So all this heuristic algorithmic method, whatever the approach you are taking, so ultimately it is going to give you the pictorial presentation. And this pictorial presentation, again, it is based on the POA algorithm. So POA is the mathematical form. So mathematical algorithm, it is going to use mathematical model to be followed to get this particular multiple sequence alignment method. So if you look at in this particular picture, suppose this is the two protein sequence we have aligned, and we try to extract the information related to these two. So if we are doing this alignment, so first the Smith-Waterman algorithm, that particular process yesterday I have explained. So it is going to follow here, and based on that the dynamic matrix, if it is required, so partial dynamic matrix, two dynamic matrix will not able to continue here. So basically that is continue, and hardly 3 to 4 gaps will be created here, not more than that. So the 3 and 4, whatever the number is required, it will be created. And now after aligning this proper way, this is the outcome or graphical presentation. It is going to come in front of your monitor. So what does it indicating? So M and M, it is matching. So this is the starting point. I and V, this is the bifurcating point. This should be V. So I and V, it should be instead of L. That is the bifurcating point. Then L, again, it is the merging point. Then K and D, again, it is the bifurcating point. G and G, a merging point. And next, as gap is there, it will be empty. But as the below sequence, the CAW, this particular alphabets are there, so it will be continue with that. Again, it will be coming to the merging point, and that means consequently T and K, these are two particular point it is matching, so T and K will be there. And then QT again diverging, P and G diverging, R and K is diverging. So this particular alignment or this particular condition is based on the POA algorithm, and that is going to give you a particular pictorial presentation. So if you looking that pictorial presentation, it will be very easy for you to understand where exactly the matching, where between the two sequences are not having any match, or these two sequences whether belonging to the two different amino acid group, or it is belonging to the same amino acid group. So this is the ultimate thing. It is going to give us best idea to understand about the multiple sequence alignment process. So this is falling under the heuristic algorithmic or category. So progressive alignment, iterative alignment, and the block-based alignment type. So I'll explain now. So how this particular process is continuing. So here in this progressive alignment method, that their global alignment process we can follow. So global alignment, how to do, yesterday I mentioned. So this is the same method of global alignment process, that is the end-to-end, that comparisons will be there, and that is going to use here in the progressive alignment methods. So here what actually we are doing, just come to this particular picture which you will better understand. So A, B, C, D, E, like the 5 sequences I have put it, and I want to compare between them. Of course, they are the homologous sequence. So once we are providing to the software, immediately they'll do the pairwise alignment. So A with B, B with C. So just so every particular sequence will be compared with the other sequence. Fine. So once we are comparing with that, we are going to create a distance matrix. So distance matrix also yesterday I mentioned. So based on the comparison and based on the that matrix value, we are going to get, and of course the logarithmic value, log odd value, we are extrapolating from it, and we are creating this kind of distance matrix. Now after looking this distance matrix, so we try to find out, our software tried to find out, so who is closest with whom. Suppose A and B, we can see the gap between them is very less. So A and B, suppose it's very closer and making one group. Now C and D, if you take the summation of this line, that score will be C and D will be nearer. So C and D is going to make the one group, and now only E is left out. So E and the E's total score is much more than A, B, C, or D, whatever is there. So E will be the out group. So E is going to connect with all other groups. As I mentioned, based on the score, A and B we are putting together, then C and D also making together, and E is the out group. So this way, this particular guide tree is creating. Okay, this is I'm repeating, this is called the guide tree. This is not the actual phylogenetical tree. So this is only the guide tree. The guide tree is going to give you idea. So this particular sequence might have very close relations. I repeat, might have very close relation, not exact relation. So now after creating this kind of guide tree, again this between A and B and C and D, as these are these two are making a group, so between them again the comparisons will be there, and this comparison will be based on the dynamic programming process. Fine. So here I never mentioned this is the dynamic programming process, just pairwise alignment process have used and try to create this kind of your matrix. So based on this matrix, we have created this kind of one guide tree. Now from this particular A and B, as it is one group, we have expected C and D should be one group. We have expected now between A and B and C and also between C and D, there will have a dynamic programming method, and by once they are applying the dynamic programming method, so here both the cases the lot of gaps will be created, and based on that we are going to get the score. Now once we are comparing it between A and B, we try to find out what are the common conserved sequences are present between these two, and C and D, once we are comparing, here also we try to find out what kind of conserved sequences are present between them. So now we are software will be giving emphasis only on the conserved sequence, right? So C and D, the conserved sequence, this is suppose, and A and B, the conserved sequence, this is suppose. Now against this conserved sequence of these two group, E will be compared, and E also will be tried to find out whether it is having the conserved sequence matching with the AB or it is having matching with the CD. Suppose here E, if conserved sequence is matching with the AB, then the E will be connected with the AB. Otherwise, if they found the conserved sequence is having matching with the CD, then E will be connected with the CD. Otherwise, if it is found that there is a no conserved sequence, there is a not matching up to a threshold with the conserved sequence between CD and AB, then the E will be connected outer way like this. So this is the very best and suitable method to creating the phylogenetical tree. And based on the conserved sequence data, again the alignment will be there, and this alignment result we are going to see in front of your eyes. So when we are doing this experiment, this alignment kind of things once we are getting. So this is the alignment kind of things we are getting that will be completely based on this particular process. So POA algorithm, and that is the dynamic programming method also using, and progressive alignment method process, that particular server or software if you're using, it is going to present the information this way. And next, this particular process actually in the practical session also you'll see, and I'm going to show you also. So Clustal is the best program which is following this kind of progressive alignment method. So Clustal, it is the URL of www.ebi.ac.uk/ClustalW. So Clustal is having two different variations. One is the Clustal W and the second one is the Clustal X. So W version is provided very simple text-based information, and whereas Clustal X, it is going to provide you very user-friendly and graphical representation. So always you look for the graphical representation, and so there will be most useful this Clustal X version. So both the Clustal W and Clustal X, they are using this particular progressive alignment method. And how basically we need to continue this. So Clustal is having, this is the symbol of Clustal. This is called omega symbol. So Clustal, when you are opening this particular software by searching the Google, so you may get this kind of symbol. So one is the Clustal, and under Clustal, this Clustal W and Clustal X will be present. Fine. So if you are searching on the Google, suppose multiples only you are typing Clustal W or multiple sequence alignment. So both the things may appear in front of your system. So you can click either on the Clustal Omega, otherwise you can click on the multiple sequence alignment by Clustal W. So if you are clicking on the Clustal Omega, so this Omega is indicating total Clustal. Fine. Here you are having the chance, or you are having the options. You are going to use the Clustal X method or Clustal W method. So this could be the home page of the Clustal Omega. So you can see here it is written multiple sequence alignment. So again, you can put the protein, otherwise nucleotide sequence here. So if you're working with the protein, let it be by default. Otherwise, you need to click over here. Here an arrow is there. So here we'll be having nucleotides or the protein. If you are working with the nucleotide, you just click on the nucleotide. So here nucleotide will appear. So now below there will be having box. These are the dialog box. So where you need to paste your sequence. Of course, the sequence should be in the FASTA format. Yesterday I talked, the FASTA format is the greater than symbol followed by the definition line, and then your sequence alphabet will start. So that particular FASTA format you need to use here. And as I'm beginning in my lecture, I mentioned, so for multiple sequence alignment means 3 sequence or more than 3 sequence definitely to be present. If you're taking 2 sequence, that will be the pairwise. And if you're taking 3 sequence or more than 3 sequence, that will be the multiple sequence alignment. So you need to paste your at least 3 sequence one after another. And you can look at here, greater than symbol, this is one sequence, then next line wherever it is ending, that is another sequence, and below after it is ending there will be my third sequence. So you need to paste your sequence over here, and you need to again provide the mail ID because the result may appear into your mail. Otherwise it will take some time to after analyzing to providing the result. So you need to provide the mail ID over here, and you need to click on the submit button. Once you are clicking the submit button, the hyperlink result will be appearing to your mail. And if you're clicking on this hyperlink, this is the result page. So in the result page, you can see that this is the alignment. So, okay, these are the between the 3 sequence, how they have aligned, and this alignment they are showing. So this is the global alignment. As it is global alignment, so it is starting from the first point, it is going to the towards the end. So they are doing the global alignment, and based on this global alignment, further I'm telling this is called the phylogram. This is only your guide tree. Okay, this is not actual phylogenetical tree. So you can look at the, by clicking on the guide tree, you can look at the phylogram, so you can understand, okay, this is that, this is going to happen. An actual phylogenetical tree, this option it is given here. If you clicking on that, so you can look that phylogenetical tree. So as here, this 3 sequence are whatever I have used to for showing you people, these are all an interferon of your 3 different organisms. So this, as these interference are having maximum similarity, 99% of similarity, so they are going to make the equal distance this particular line and showing the phylogenetical tree. So as a beginning, I mentioned the multiple sequence alignment help us to understand how to create the phylogenetical tree. So just here one approach, this is the phylogenetical tree, but again getting this phylogenetical tree, lot of your algorithms and lot of process are there. I'll be explain next next to it. And if you're typing directly cluster W in your Google, so you are going to get the cluster W, and you are opening the cluster W, and this is the server of it. Okay, homepage. So here you can, if you are clicking, you can the cluster and different mode of cluster also you can get it here. And you just paste your sequence. Once again, here 3 sequences are required, as it is the multiple sequence alignment. You can paste one after another, and you can just below you can go for these are the, if you scroll down after pasting the sequence, if you scroll down, these are the another options will come. So many options, it should be leave it by default, otherwise you can change it. So some scoring pattern is also mentioned here, we can follow the gap penalty is 10.0 and extended gap penalty is 0.1, or we can use the which matrix like Blossom 62 PAM, what kind of matrix I want to use to get the score. By clicking here, you can change that particular matrix also. And after selecting all these criteria, you just click on that executive multiple alignment. So if you are clicking on it, immediately you will be getting this kind of result. This is the first page, and top portions of your result. Here the what are the sequence you have provided, what are total number of alphabets are present, etc. It will be mentioned with all the 3 sequence, and matching percentage also between the 3 sequence also to be mentioned there. And if you scroll down again, you are going to get this kind of alignment. Fine. So this is the alignment is indicating that where is having between the line wise, where is having conserved sequence, semi conserved sequence, etc. Wherever you are going to get the single dot, that is indicating that is might be replacement happen between the other group of sequence, other group of amino acid. Wherever double dot, that colon type of form you are getting, that is indicating that replacement happen between the same group. And wherever asterisks will be provided below this line, so that is going to indicate perfectly matching that particular alphabet in each sequence. So this way you need to understand that what it is going to tell you and what kind of information is going to provide you. And from here also you can click on that different type of phylogenetical tree, what you are looking for. So you are having neighbor joining method, cluster, you are having Pymol cluster method, we are having fast tree method, different methods are there, and we always look for the neighbor joining method. So we just click on the BioNJ, that is the neighbor joining method. So if you are clicking on the neighbor joining method, you will be getting the tree. Otherwise, you can select any one of this particular tree mode, and ultimately you'll be getting in your tree. This is the top part, and if you scroll down, this is your tree. So look at here, this is the phylogenetical tree, and it is going to indicate these 2 are the cluster or the group, and this group is connected with the third one. That means AB, if we consider A, B, C, suppose B and C is a one group, and it is going to connect with the group A. So group A is the outer group, and group B and C, and if from this particular phylogenetical tree we can predict this group A. So this A is the rooted one, and B and C is the branch one. So this from this root, this B and C have created. So I'll be coming to this concept afterward, and this is the different tree changing pattern. Any one of you can click, so you can understand that what kind of scaling pattern they have used. So if you are clicking any one of these blocks, like without branch length, with branch length, etc., etc. So if you are clicking, you can see that scales are moving. So here, this particular dialog box, whatever is given, you can select any one of it, and you can see how this particular scale is moving. So anyhow, this is the way how we are using the cluster W or the cluster X for using the multiple sequence alignment, and from there try to draw the phylogenetical tree. So from cluster directly you can go to the phylogenetical tree. Otherwise, only for creating phylogenetical tree, there is having very other kind of open based software like your T coffee, and you are having that cluster P. So these are the different mode also it is available. So they are also, you can use the similar way. First you need to create the multiple sequence alignment, and then from the multiple sequence alignment you need to move for the making the phylogenetical tree. So this is the way basically cluster is working, and the cluster is following this particular pattern before projecting the result. And second important, what I was telling, peak of a DB cluster, then POA, Preline, etc. So among that, peak of is available freely database, but sometimes they need to restrict something because of their modification of the software. Always you may not able to use all these, but the cluster W and the cluster X, which you can avail throughout the year, there will be no problem at all. So next algorithmic method, that is also based on the global alignment process, that is called the iterative alignment. So I am coming directly to the iterative alignment. So here what is happening? So suppose these are the sequence I have provided. So based on the sequence, just by looking the length of the sequence, they are going to make the group. I repeat that by looking the rest length of the sequence, they are first initially making the group, and this way they are creating the guide tree. Just previously I mentioned the guide tree. So they are creating the guide tree here. These are indicating this group is connected with this group, etc., etc. Now after creating the guide tree, again they will be trying to align. So whatever the groups are there, between the group, again they will try to align, and after once they are aligning it, here they are following the actual dynamic programming method here also. And based on the dynamic programming method, so they found, suppose A is very closest to the C when they are matching or pairing, but initially guide tree it was A with suppose X, it was having a group. So that this way, after doing the dynamic programming method, they are going to actual segregate based on the scoring pattern. So now after getting segregating, again they will be aligning. After aligning again among them, they will be creating the multiple sequence alignment procedure. That means again with all them, they will follow the similar guide tree method. Now the guide tree, it is going to give you idea exact way how A will be connected with the B, and C how it will be connected with the AB, etc. Now in this case, when they are going for doing the alignment using the dynamic programming method, so here whatever the score they are going to get, the score they will try to tally from before creating the guide tree, what was the sequence. From this sequence also they are going to find out the scoring pattern. And here after doing the dynamic programming, here whatever the score will be getting, the score will be tallying, and which score is the better, based on that it will be considered that this is the best alignment process. And of course that is depending on the POA algorithm, that means graphical mode we are going to get, and that is of course by chance those sequences will be present in that, and there is a statistically it is highly will be significant. So here outer iteration it is indicating after doing this alignment, we are going to compare with the this original sequence alignment. So this way this particular method is working, and of course this method is useful for making the UPGMA method. I'll be talking next part. So how this particular UPGMA method and all this particular process is working. So when you are using the software, we do not know how the software is running. Only we are would know just how to paste the sequence and how to click on where and what to get the result. But why the software is running this way, what logic is there, what kind of algorithm basically is going to follow there, if that is understanding is very vital things in any kind of bioinformatics. Just not it is going the analysis, I know everything, it is not like that. Okay, biology with informatics, it is very important. And informatics means you need to know the algorithm, you should have the basic concept on the computer science, otherwise bioinformatics nobody will be able to understand. So anyhow, this is the particular process it is going to use. And the third method, that is the block-based alignment method. And you can look at here, block-based alignment method is basically based on the local alignment process. So here the Dialogin 2 and all the software or Matchbox we can use for it. So actually the block-based alignment method, which is considered as the local alignment process, it is going to work in this way. It is going to work by creating the profiles. Okay, so for creating the profiles, what you have to do, the sequences, whatever you wanted to compare. So first that you need to do the psi blast, and by doing the side blast, you are going to get some kind of informations, and those informations you are going to use here. So what is going to happen? First, you are doing your query sequence, and with the query sequence, you are comparing your database sequence, and you are getting the blast output or the blast result. From the blast result, yeah, by looking the E-score, yesterday I explained, by looking the E-score, we need to select that these are the sequences are having perfect matching. So I can take these are the sequence for creating the phylogenetical tree or to continuing the other work. So those sequences we are selecting out of the blast result, and those sequence which are having the E-score maximum matching. So those are called the, of course, homologous sequence. Until unless it is homologous sequence, no E-score will be perfectly matching, or it will be not be nearer. So you need to understand that what are the sequence I supposed to collect from after doing the blast. So those sequence of homologous sequence you are selecting. Now by taking this homologous sequence, you have to do the multiple alignment. So those homologous sequence you are taking, you are creating the multiple alignment process, and by doing the multiple alignment, you are creating the profile. So what does it indicating this profile? Profile is indicating how you are comparing A with B, you are comparing B with the C, you are comparing A with the C. So this way, after comparing and you are getting the logit values, whatever, so that is telling your profile score. So this profile you are creating. Now, after looking this profile, you will understand that A is whether it is connected really D, or C is having really relation with the E or not. So if you found by looking the profile score, A is connected with the D, means the A score and the D score is very nearer, then you need to take separately A sequence and the D sequence. Again, you need to do the pairwise alignment, or you can do the again the blast. Again blast means this A sequence you are taking and you are considering D is the very closer to it. So A and D, any one of the sequence you need to take, and you need to go for again normal blast searching. So normal blast, initially you need to do the side blast, and now you need to go for the as it is the protein sequence, you need to go for normal protein blast. So by doing the normal protein blast, whatever the protein databases sequences are there, which is similar with your A or the D sequence, it will be matching, and again it is going to give you the proper additional homologous result. So again, by looking the E-score, you need to consider those particular sequence. Those sequence you need to take. Again, you need to create the new profile. So now this new profile and the old profile, if it is somehow if it is tallying, then you need to consider in this particular first profile, whatever the sequence you have created or alignment, whatever created by the software, that is only correct one. And if you found here this new profile sequences are not matching perfectly, if this sequences total score, it is deviating from this original profile score, then again with this from here you need to consider, suppose now C and E is very closer. Now either C or E sequence you need to take. Again you need to create the blast. Again you need to get the additional homologous sequence. Again you need to create the new profile, and that profile again you need to connect with that. So actually what is going to happen here, by creating the profile, we are going to understand that whatever the sequences are present in the database, whether those sequences are perfectly matching, and based on the matching, we are extrapolating that information. These sequences are very closest to each other. So that is the meaning of creating the profile. And of course, this profile creating we can able to use again by cluster W. So I have just mentioned that cluster W, how to use it. So if you are opening the cluster W, the last here, if you are opening the cluster W, so this is the homepage of cluster W. Yeah, if you are opening the cluster W, and then there will be having your options. So if you are clicking here, so which mode you are going to get the result, you just click on that particular mode result you will be getting, and beautiful way you can able to see the multiple sequence alignment. And I mentioned here the one asterisk should sometimes to be present, or it could be colon, or it could be single dot. So you are going to get the idea from there, and that is going to tell you whether your profile, whatever you created, the profile is perfect or not. So this way, these particular process are continuing in by doing the side blast method, and to create the profile, this particular activity is possible. So the next approaching method, that multiple sequence alignment, that you came to know what kind of what kind of algorithm basically are using, and how these algorithms are helping to understand that sequences are perfectly aligned and the sequences are really having any kind of matching or not. So the main aim for multiple sequence alignment, of course, to creating the phylogenetical tree, and to understand whether these sequences are having evolutionary any connections or not. Even if you are comparing the proteins, you try to find out that what kind of conserved sequence, domain wise what kind of conserved sequence, motif wise what kind of conserved sequence it is present among those particular homologous protein sequences. So our main theme for doing the multiple sequence alignment to create the phylogenetical tree. Fine. So I am going to talk now on the phylogenetical tree. So before coming actual how to tree should be created, you need to get the basic idea about the phylogenetics and how on the phylogenetical tree. Okay, so tree means what are the parts should be there, what is indicating what, that is I'm going to talk first in the phylogenetical basis. So phylogenetical tree, it is a kind of it is a tree like presentation. Okay, so if you are taking the few sequence, if you are doing the multiple sequence alignment, and with the respect to the multiple sequence alignment, if you are going to get some kind of picture, the picture will be tree like of picture. So phylogenetical tree, it is going to indicate the relation between the phylogenics. It is going to tell the evolutionary relationship among the various organisms, whatever you are going to understand that how in the evolutionary scale, how the evolutions happen and what way the evolutions happen. So both kind of concept basically you are going to get from this phylogenetical tree. So phylogenetical tree, if you can take for creating the phylogenetical tree either DNA or RNA sequence, or of course you can take also the protein sequence. So this is the normal picture of a tree, and this tree is indicating this is having certain kind of line, and on the top of the line we are putting the different organisms, and we are establishing the relations between this organism with respect to the evolutions. So in this phylogenetical tree, we need to understand certain kind of terminology. So first terminology is what is called the branch. So branch, if you look at this tree, so this is a particular point from where this particular line you have started plotting or started drawing. So this particular, from this point, whatever the lines you are drawing, so these lines are called the branch of the tree. And this point from where you have started creating the line, this is called the root part of this tree, and this root is going to indicate this is the common ancestor, whatever the organisms are present on this particular tree. So you this is the branch and this is the root part, and from the root, this whatever the branches you have plotted from the branch, if you are taking a particular point, again from there if you are creating the some line, so this line also is going to indicate these are also the branch, but the point from where you are again creating the branch, that is called the node point. And on the top of the node, that means whatever the lines you have created on the top of the line, that particular position is called the tips part, and the tips part you need to put all kind of organism. So your main aim to find out the relation between all these organisms, suppose lions, leopard, jaguar, tiger, domestic cat, etc. So what kind of relations they are having and how they have evolved, evoluted, and finally we can able to understand their evolutionary pattern. So this is a normal presentation of the tree. Now you need to remember there is no specific way to create the particular tree. I repeat, there is no specific way, means there is no specific topology. That topology is going to tell this is the only way to create the tree. No, there is no topology as such. So anybody can create the tree by any method, any way. So there is no proper topology of it. So same things whatever I have mentioned here, lion, leopard, jaguar, domestic cat, tiger, etc. So how I am making the tree here, I can make the tree, same tree in this way, I can make the tree this way also. So that means there is no proper topology to explain the tree. But always in the books, it will be given this kind of picture. So all of us suddenly having in our mind that this is the only picture of the tree. So phylogenetical tree means this is the only tree. No, it is not like that. So in a particular tree, if you look at the top part. So that whatever the organisms you're putting over there, so that is called the terminal node part of this particular tree. And from the main branch, whatever the the branching is starting, that is called the internal node. And this internal node, whatever the organisms you are placing, that organisms are having certain kind of relations. So here again, this after placing the organism, we can think the tree in this way. This is the clade or monophyletic tree, or paraclad, or your your lineage tree. Okay, so monophyletic tree or the cladding tree, if you look at here, this particular part, B and C is closest with the A, but D and E, it is present to the completely other node. So D and E and ABC, these two having some kind of relations, but in the closest relation, if you look, that definitely B and C will be having A closest relations. Fine. So D and E together consist of one clade, whereas ABC together is creating the another clade. And there is a relations between these two clade. What is the relation? They are originated from a single particular point, and based on their evolutions, they have ultimately reached to that particular jaguar, leopard, all this particular organism. So this is the concept of the clade. And a paraphyletic tree, if you look at here, paraphyletic concept. So paraphyletic is telling B, C, and D how it is related. That is the paraphyletic relations. Whereas cladding or monophyletic relation is telling ABC is one group and DE is another group. That means DE having very closest relation, so they are the monophyletic group. ABC is having closest relation, they are the monophyletic group. But if we compare this ABC with the DE, that is going to tell you that is the paraphyletic concept. And as I mentioned, there is no proper topology, so anybody can present the tree in a different way. And a dichotomy, it is telling how this particular internal node we are joining with each other. That means here B and C is one clade, where in this clade that outer clade will be A. So how we are joining A with this B and C, that is going to indicate a particular line, and based on line we can sometimes tell this particular tree is the cladogram. So cladogram is basically indicating how you are connecting between the all node in a particular tree. So sometimes the polytomy concept also comes. So whatever the sub branches is coming from there, again some organisms, if it is arising between the two terminal node, then we can tell this is the polytomy. And finally dichotomy, I mentioned how we are connecting this particular all organisms with in a particular clade or particular group. So based on that we can tell the dichotomy. So again some software and can present the tree in the again two different mode. One is called rooted tree mode and second one is called unrooted tree mode. In the unrooted tree mode, look at this particular picture where there is no original origin concept here. So AB, you can just understand there is a having relation between the AB and the CD, and of course there is having a gap. So A and B, it is very closest, and there is having evolutionary gap, and C and D also is very closest. And based on the A and B, if we consider after this evolution, C and D has created. So this kind of picture is only going to give you idea just A, B, C, D is having a relation. That's all. Who is the ancestor of whom, whether from A this BCD has created or from the D this CAB has created, that we are not able to understand from here. But if you look at this particular tree, this is having a particular root, and from this root suddenly the A, B, C, D all have created. So that means this kind of picture is called the rooted tree picture, and this type of picture is called the unrooted tree picture. So rooted tree picture always will give you idea who is the originator or who is the common ancestor for all this particular other organism. So the software whatever you use, basically we try to look for the rooted tree, because rooted tree is going to give the best information to you. And when you are talking about the phylogenetical trees, a lot of common terms will come: monophyletic, paraphyletic, already I have explained. So you need to understand those definition. And now I'll be talking about the phylogenetical tree construction method. So how generally we create this kind of phylogenetical tree. So that is having, this is the particular flowchart basically we can follow when we are using this experiment. This particular flowchart only you need to follow, and you need to get the tree. So first thing is the selection of the organism or a gene family. So among which organism you are going to establish the relations. So that organism you need to select first. Otherwise, if you wanted to relate with a certain gene family, so you need to think that from what are the different organism the similar gene is present, and you need to get the idea about that particular gene and the gene sequence, and with the gene sequence you can continue your phylogenetical tree and multiple sequence alignment as well as the phylogenetical tree making. Like that, if you talk about the protein, so that protein, whether it is found for the all animals or not, if it is found to the all animal, so you need to get the protein sequence from each animal, and then only you can compare with it. So this is the first step, and after getting that, you need to choosing the appropriate molecular marker. So this is another very crucial and important parameter. So I beginning itself I mentioned just anything we cannot take and create the phylogenetical tree. Anything we cannot take and just do the multiple sequence alignment and create the phylogenetical tree. No, it is not possible. So when you are before starting your experiment, you have to be definite enough. So what particular sequence is supposed to take, and what particular protein sequence also is supposed to take, whether that protein could be useful as a molecular marker or the gene sequence to be used as a molecular marker. So what are this molecular marker? Molecular marker, it is lying in the sequence level itself. The molecularity should be there in the marker, and by looking that particular marker, you will get the idea. Yes, I can use this, and I can continue the my phylogenetical analysis, and I can create whatever the tree is going to accepted by everyone in this world. Okay, so that idea you must remember. So molecular marker you need to select. So simply just you can take one protein and you can compare with other, and if you are telling, yes, I have created the phylogenetical tree and that is the best result. No paper will be accepted in such a way. Okay, so we need to give the emphasis on the molecular marker. So many molecular markers are there. If you talk about the protein or gene sequence, these are the table. This is the going to indicate this is what kind of molecular marker basically we use for creating the phylogenetical tree. Means multiple sequence alignment followed by the phylogenetical tree. So for example, the gene sequence like EFTA, that is the elongation factor type A having role in the protein synthesis. So this elongation factor found in all kind of organisms, and eukaryotic organisms we know that are having a lot of elongation factor, transcriptional factor, translational factor, even DNA replication enzyme, and all kind of eukaryote must have that particular enzyme or that particular protein. So we need to give emphasis only those particular marker. Okay, so like RPOA gene, that is encoding the alpha subunit of the RNA polymerase. So that is with respect to the prokaryotes. So we can take that particular part. Otherwise, DNA, that is encoding the DNA polymerase. Or if you look at the LUGS gene, that is going to give the luminations. Okay, luciferase, that particular gene. You know that luciferase gene, if it is present, if it is going to give the glowing light. Okay, so you need to think of which particular marker supposed to use. The marker, whatever is supposed to use, that marker definitely is going to keep the molecular level informations, and those marker definitely should be present to the all organism, and that marker should not go for complete different type of mutational process. It should be avoid to various type of mutations, and that's way it is going to give the proper molecularity or molecular level informations, and you can select those in molecular marker only to your research purpose or to doing your exercise. So you need to select the molecular marker. Then if it is possible, if you're having only one single gene sequence and you are having some other activity to continue with the gene sequence, in that case you should not lost that particular sequence, so that you can amplify that sequence by using the PCR. You can make the multiple copy of it, and after that you can go for the actual alignment activity. So once you're doing the alignment activity, evolutionary model you are creating based on the evolutionary model. This evolutionary model is indicating this is the guide tree. So you are creating, then you are creating the phylogenetical, you are doing actual phylogenetical analysis, and based on the phylogenetical analysis, the tree is going to construct and it is going to appear in front of you. So the tree, whatever is going to appear, that is called the phylogenetical tree. So this is the fundamental protocols always we follow to understand related to the gene and the protein, what way this phylogenetic tree should come and how the phylogenetic tree should looks. So here I am telling that the nucleic acid, otherwise amino acid, you can select as a your sequence to continue this kind of multiple sequence alignment of the your phylogenetical tree. But you always need to remember when you are creating the phylogenetical tree, the modern basis of to creating the phylogenetical tree to give the emphasis only on the nucleic acid, because nucleic acid directly the gene composition directly can relate the phenotypical and genotypical appearance of that respective organism. And the gene level, if you are tracing, if you are comparing directly, we can come to the species level and we can relate the correlation with the among the various species. Fine. But if you're taking the protein, all that protein never ever will help you, or it will give you that kind of phylogenical tree which you can correlate to their gene level. If you're taking the various protein, example like hemoglobin, if you're taking from the different organism, the protein sequence, whatever you have taken, it is going to give the idea only the evolution of that particular protein, wherever the mutations happen, what kind of conserved sequences present, all those. But it is never ever will able to correlate with the gene level. Fine. And you will never ever able to correlate the evolution pattern of the entire organism from whom you have taken that particular protein sequence. So modern phylogenetical study, always it is telling you give emphasis only to the gene level, nucleotide level, not to the protein level. Fine. So you need to understand why it is telling like that. And this kind of whatever the marker we are using, that marker must have certain kind of property. So I'm telling these are the few important property, like it should be present in a single copy. It should not present in the multiple copy to the any organism. This is the first important vital point. Second point, and this alignment should be very easy. That means the sequence, whatever you have taken, that should not be too much lengthy. So it should be optimized length like 200 to 300 or maximum 500 nucleotide should be there. And if we amino acid related, if a protein level, if you are talking, that should be maximum 300 or 200 or 500 maximum amino acid, because too much amino acid if it is there, so it may create some kind of trouble to creating the phylogenetical tree, and the software also sometimes to be puzzle, and it will take a lot of time to provide the phylogenetical tree, and the researcher will not be having so much time and patience. So the alignment should be easy, and then region, whatever you are selecting from the gene, that should not be ambiguous. That means if ambiguous means the mutational process already I mentioned. So hemoglobin of human and hemoglobin of chimpanzee, if it is having 90% difference, then that particular protein you should not able to consider. So that kind of ambiguity should not come to your selecting marker. And substitution rate should be optimum. That means if you are taking the nucleic acid sequence, there also the substitution A is replacing by purine or pyrimidine. Maximum should not have the maximum rate of substitution. It should be moderate. Okay, suppose after 30 nucleotides, some one positions you can get that kind of changes, or after 20 nucleotide again you can get some kind of changes. The changes should not be there continuously, like one after another. That kind of changes should not be there. So that substitution rate should be optimum. And you can able to design the primer, because in the phylogenetical tree means multiple sequence alignment I was telling, another important criteria and another important outcome of multiple sequence alignment to create the common primer and to amplify the all kind of gene sequence. So that kind of provision must have with your marker gene. So you can create a single primer to amplify all. And too much of base variation among the taxa also is not going to accept it here. So for that, for bacteria, for eukaryote and prokaryote, there is a concept. So eukaryotic microorganisms, we can use the 18S rRNA ribosomal gene component or gene sequence, and for all kind of bacteria we use the 16S rRNA gene sequence, because that particular sequence we know that they not have much variations, and evolutionary it is very conserved. So if we take this particular sequence from various taxa, easily we can able to establish the relation between them. That's why such kind of 16S or 18S sequences we are giving the preference. So we have to remember when you are selecting for your research purpose, for your different purposes, to create this kind of phylogenetical tree, or after doing the multiple sequence alignment, what kind of gene sequence, protein sequence you have to select, why you have selecting that, all explanation definitely you need to mention in your project, as well as you need to mention in your thesis. So you got the idea that phylogenetical tree basically how we used to create, and what kind of information the phylogenetical tree should have. Now we need to think that what kind of model is supposed to use for choosing the phylogenetical tree. That means what is the your process to create this construction process of this phylogenetical tree. So molecular phylogenetics, now I am going to talk. So far I gave the idea, so what way it is working, etc., and how we are creating, what kind of marker supposed to be used, and how supposed to going to use, what kind of information basically you are going to get. Now we need to understand that when we are using the software, what kind of algorithms and molecularity they are following. So this is the overall process. So you are providing a sequence and you are doing the multiple sequence alignment, and then software, if it is found is there is a strong sequence similarity, then it can go for the maximum parsimony method to create the phylogeny. Otherwise, if they found there is not having too much similarity, then they can go for again recognizable sequence similarity pattern. So if there also they found there is a not having too much recognizable sequence similarity pattern, then they can follow the maximum likelihood method and finally create the tree. And if this second stage, if they found yes having somehow recognizable conserved sequence among them, so then they can create for distance method of tree. So ultimately this is the statistical protocol and statistical model, or this is the actual programmatic protocol, what and how the software is following, and what way we can select which tree is the best one. So distance tree based method, the first approach is the distance tree based method. In the distance tree based method, so whatever the organisms we are selecting to establish the phylogenetical relation between them, we are taking these assumptions that all the organisms, whatever the sequence are we have taken, so the sequences are independently have evolved in this environment, or independently evolved among them, and every sequences are having equal chance to act as a specific homologous sequence. So these particular assumptions we are thinking, and based on these assumptions we are taking the two different approach. One is the clustering method approach, and second one is the optimal based approach. So clustering based approach here try to make the cluster by using the UPGA, that is the UPGA stands for unweighted pair group method, using the arithmetic average and neighbor joining process. So this is most of the software will try to follow this UPGA method. And second latest method, that is the optimal based algorithm method, which is compare many alternative tree topologies. So whatever the base topology, so whatever the topology is possible after getting the relation between A with B, B with the C, C with the D. So which is the base topology, which is going to give the best information, that topological tree they are selecting and they are presenting in front of you. So both this method is the clustering based method. So cluster, that means the group is going to create by using this particular algorithm. So in the clustering based approach, the first is the UPGMA. So I'm coming directly with the example. Suppose these are the sequence we have plotted one after another, 5 sequence, and we are every sequences are having 20 alphabets, and we are going to compare between them. So how we are comparing? 1 with the 2, 1 with the 3, then 1 with the 4, 1 with the 5, 2 with the 3, 2 with the 4, 2 with 5, like 3 with 4, 3 with 5, like that we need to proceed. Fine. And once we are looking with the first point, if you're looking between the sequence 1 and 2, so where is exactly the differences we can found? Here A is matching, and here A and T, that is not matching, and rest of the part we can see it is the matching. Fine. So as only one particular point it is matching, and if it is compared with the between 2 and 3, you can see here is having gap, here is not matching, and then as here it is having gap, so this gap already introduced, so definitely 2 and 3 it should be closer, as it is only single matching, but the gap also we have considering here. Like that we need to, by looking manually, if we see, so we can able to understand why and how that 1 and 2 sequences are very closer. So by looking manually, if we think 1 and 2 sequences are very closer, we can join them by this way. So we are putting 1 and 2 is very closer. Now, if you are going with this manual process with the 1,000 or billions of nucleotide or 500, 600 nucleotide, you will be very much puzzled, and this manual process is not possible. So that's why we have created a statistical protocol. So in the statistical protocol, what we are following, first we are trying to get the idea this whether 1 and 2 are having relations or not. So yes, if it is they are having relations, we are putting both of them together. And now we try to establish with 1 and 2 what is the relation with the 3, and 1 and 2 together what is the relation with the 4, and 1 and 2 what is the relation with the 5. That means initially 1 and 2 we are putting together. That means we are creating a clade, we are creating the group. So 1 and 2, if it is putting together, then we are considering this 1 and 2 with the 3, what is the relations. So 1 and 2, this is the one clade. We can found 1 and 3 is having relation. This mathematical explanation or derivations are not going, because this is not the correct platform and the time to explain that. Just I'm giving the idea, so how we are progressing or the software, how it is doing its function. So we are here along with the 1 and 2, we are creating the relation with the 3, or along with the now if we found 1, 2 and 3 are together, then now with 1, 2 and 3 is one group, and what kind of relation it is there with the group number 4. So that way you need to progress. So 1, 2 and 3 are now we have got the idea what is the relation 1 and 2 together, and next is a 3 is very closer to them. And similar way we can progress, which is the relation with the 4, and finally what is the relation with the 5. And that way if we are getting this kind of tree, so this tree is called the arithmetic average tree. Fine. So arithmetical expressions we are using, so it is called the UPGMA method. I repeat, this is called the UPGMA method, that is the unweighted pair group method using the arithmetic average. So this is the one of the important aspect to creating the actual phylogenetical tree. Now this is the tree I have created. The same tree you can plot by this way. So I got the relation between 1 and 2. So instead of writing here this way, you can put here 1 and 2, or you can put here 1 and 2. Finally 1, 2, 3, 4, 5, what relations you got, you can place by this way, otherwise you can put this way, otherwise finally you can put this way. Fine. So ultimately the tree, what topology we are getting, that is all cases how we are exhibiting, all cases it is having the same meaning. That means whether you are presenting with the line, whether presenting in the group this way, that doesn't matter. That's why beginning I mentioned there is no proper topology for any kind of tree. Only thing is whether the tree is rooted or unrooted, that is the fundamental issues. And next is the neighbor joining method, that is also clustering method. So in the neighbor joining method, so basically depending on this particular equation, and this equation is telling that actual distance and the transform distance value we need to calculate. Fine. So the actual distance value and the transform value, if you are getting, the transform value will help to understand that this one group how will be relate with the other group. Okay, so that's why we are creating the transform odd value, and this is having mathematical equation. So in this method what we are doing, we are thinking one thing I forget to tell, if you look at this particular picture of the tree, so here almost all the length are somehow is not equal. Here this length and this length is equal, and this length of course is not equal, but some part of it of course it is equal. So you need to understand what could be the branch length. Okay, so when you are exhibiting the tree, presenting the tree, there should be a branch length. Here I mentioned in the in this practical approach, there is mentioned the branch length. Okay, so you need to understand how this branch length are creating. So I'm going to tell right now. So we can see this is having some particular kind of number. Okay, so this number, this is called the branch length, and what could be the length of this line and this line, and how much distance should be there between these two, and what point exactly this root will be connected. So that I'm going to tell it right now. So in the this particular method, that is your neighbor joining method, that is also depending on maximum likelihood process. So this is called calculating the odd values as well as the transform odd values. So this is again in the previous example, suppose we got this kind of result. Now, before continuing this particular process, so what assumptions basically having in this method or in this algorithm, that whatever the taxa we are going to compare, all the taxas are having equal potentiality, and that's why all the taxas are having equal chance to mingle with other and having equal chance to make the group with other. Fine. So that's why first what we are doing, suppose A, B, C, D, these are the four taxa we are considering and try to establish the relation between them. So first we are creating the start tree. Okay, this is the tree. This is called the start tree. So you can see here A with this particular crossing point, A, B, C, and D all are having equal length. So we are giving emphasis first as per the assumptions all are equal. So now if we found as per the calculation of R and R value, if you found the A and B, it is very closer. So we are putting them A and B together, and this A and B we are joining with a particular vertical line, and then on horizontal line we are taking and we are joining A and B. Now we are considering this, suppose between this A and B, this particular vertical line distance is U. Now what could be the distance between A to U and what will be the distance between B to U? So if you are calculating it, automatically the length of this particular line will be shortened down as per the calculation result. So we are following that. So we are getting between A to U, we are having 0.15 scale distance, and between B to U, it is having 0.25 scale distance. Like that. So A and B, this relation we got. Now we need to understand next group C, how it is connected with A and B. So we are calculating the R and R dash value. So and we are getting the C with the particular how it is connected between the C to this V. Okay, this distance all are placing in one particular positions. What is the distance between the C to V? If you are thinking this A and B, it is connected with the some distance with the V. So what will be the distance between the C and V? As per this mathematical calculation, we are getting 0.15. So in this particular position, you can look at here the length of each line. Okay, length of each vertical horizontal line, it is going to different. Okay, the length of the line is going to different, and that's why this kind of tree making is giving much more information about the evolutionary process. So how far this with A and B are very closest, okay, no problem, but A's evolution, the B has more evoluted than A as per this line, and C is somehow it is connected with the A and B group somehow, but the C is having some outer your evolutions, and based on the outer evolution, the C is somehow got modified, but C is maximum matching we can found with the A and B group. So this way we are creating the final tree, and all this kind of tree is going to tell you the branch length and the branch pattern. So branch pattern, it is telling who is connecting with whom, and branch length, it is telling what should be this 0.15 this length difference, 0.25 this length difference, and 0.15 again this length difference. So this way this method is much more useful, and when you are presenting any paper, when you are putting any kind of research for your publication purpose, definitely they will ask provide this particular branch length, how much you have used for the tree, everything. So this is the way how software is calculating, and I have mentioned here in this practical. So if you are wanted to know the branch length, if you wanted to see without the branch length and the level and without the leaf level, so you can click on this box and you can see beautifully that length of this particular lines is changing. Fine. So this option, it is there for the software, and software basically following this particular algorithm. Next method is the maximum likelihood method, which is also known as the Fitch's parsimony method. And this Fitch's parsimony method, it is completely giving the idea based on the alignment score. So what exactly how we are following? So look at here. Suppose we have aligned this particular four sequence, and of course this is a multiple sequence alignment. And among this sequence, as per the Fitch's this parsimony method, they are having the two different assumptions. So first assumption, it is telling the site should occupy by at least the two different type of nucleotide. So if we compare one line, so at least two different type of nucleotides should be present in this particular line. And second important point, the two different nucleotides should present at least twice at a positions. Duto jayega, two different positions, definitely these two nucleotides should be present. So if you look at this first column, here all A, second column A, then 3G. So 2 times it is not repeating. 3 CGAA, GCAA. So G is also not repeating 2 times. C is also not repeating 2 times. Like fourth also it is having same kind of conditions. So only position number 5, position number 7, and position number 9 is having similarity as per these particular features assumptions. So here is point number 5. You can see the two alphabets are there, and both are repeating twice. Like 7th also having GG, CC, and 9th is having AA, GG. So we can consider 5, 7th, and the 9th. These are the positions, and we need to create actually from this features parsimony. It is telling. So based on this kind of information, so as I mentioned, this assumption, based on this information, they'll be creating n number of tree topology. And after creating the tree topology, we will try to find out which particular tree is going to give the maximum information. That maximum information tree they are going to consider. So how they are creating? So first example, look at here. If we look at the position number 7, so it is written 3. I mentioned it 7. So it is, if you look at the position number 7, okay. So this is the position number 7. Here 2G and 2Cs are there, right? It is written wrong. So here position number this should be position number 5, where G this G one will be matching with the A, this G will be matching with the A, then AA also should be matched. So this way they are going to create the tree. Okay, so various tree topology they are creating. And if you look at the tree topology 1, tree topology 2, and tree topology 3. If you look at this one, so how many informations we are getting from here? AA is matching, AA is matching, GG matching, GC is not matching, so 1 information, and here GA is not matching, so 1 plus 1, 2 informations we are getting. So tree number 2, here also we are getting this GA is not matching and CA is not matching, so 2 information. Tree 3 we are getting from here, GA is not matching, CA is not matching, the rest of the things are matching, so here also we are getting the 2 information. Like next position, suppose with the 5 if you are doing, you come to the 7th. So similar way if you are doing it, so you need to find out whatever the tree is possible to extrapolate from the tree, how many informations you are gathering. So that information you need to note down, and based on the information you need to create this kind of summating table. So tree 1 with respect to the sequence 5, we got the only 1 information. Request sequence 7, we got the only 1 information. Sequence 9, only 2 information. Like that, sequence 2 and sequence 3 we are taking, and finally we are summating in all, and we are getting this value. So 2 plus 1 plus 1, 4. So here 2 plus 2, 4 plus 1, 5. Here triple 2 means 6. Now look at this particular table. You need to consider that which is the lowest number. That means lowest information from which particular sum we got it. So that particular tree is the best connecting tree. So suppose this is the 4th, it's the lowest number. So this 4th positions, this 4th, that means position number 1, the tree, position number 7th, and position number 9, it is going to give you the perfect informations. So if you look at this with the respect to the 3 alphabet, same example, you can take the GGA or GGG first. So GGA and ACA, GGA and ACA. So one with the 3 first they are going to compare. And with respect to these 3 alphabet, what kind of changes are present? So the changes are making here GG, A, and here is having ACA. So G and A, one not changing, means not matching. G and C is not matching. A is matching. So 2 information like that. This way the information they're gathering and try to write down the summated changes number. So here 2 plus 1 plus 1, that is the 4. Second, if you are taking the GGA with the GGG, if you look at here, GGA with the GGG. So in that case also, what are the different informations we are getting? So that is coming 5. And third, between the GGG and AGG, what are the informations we are getting? That is coming 7. 1, 2, 3, 4, 5, 6, 7. It should be 7. So 7 informations we are getting. So now this information wise, it is going to tell you this 4th, that is the first tree, that is the relation between the first, this particular sequence, and followed by these are the best one to creating this phylogenetical tree. So this way the features parsimonies are working, and finally this kind of tree, lot of topologies they are making, and the base tree information they are gathering, and the tree is going to project it in front of your monitor. So this is the all about how we are creating the tree, what kind of protocols are following, and what are the various algorithms to be followed, and finally we are going to get the tree. So now you need to understand that evaluation process of the tree. That means what kind of statistical evaluations are going on. So until in the bioinformatics, the software is working based on the statistics. That's why the result, whatever we are getting, we are telling this is the perfect one, as if you are able to follow the perfect rule or correct rule. So the tree, whatever you are getting, how come I'll tell this is the correct tree? So for that, again, the software is continuing with the phylogenetical tree evaluation process. So here, for creating the evaluation, bootstrapping method is the best method, and bootstrapping method created the tree is well accepted for all over the world now. And bootstrapping method, basically we can see on a particular software. If you look at my monitor, here is having one mega X is written. So this software, if you are using exactly the bootstrapping and all kind of statistical valuations we can do. Whereas this cluster W and whatever the things are mentioned, and cluster lakes, whatever the free softwares is there, those free softwares will never, only they'll be giving you the phylogenetical tree, but it is not going to do the bootstrapping activity or the statistical parameter valuations. So there will be having a big question mark once we are putting by using the free software this phylogenetical tree in your research. So always you need to follow certain kind of purchasable and useful software once we are doing this kind of genetical analysis or protein related multiple sequence analysis and creating the phylogenetical tree. So what basically the bootstrapping method is telling? So bootstrapping method, it is basically try to find out the by non-parametric and the parametric method, it is going to establish the relations that whatever the tree we have created or the softwares have created, that is the corrected one. Okay, so what is the parametric bootstrapping method? In the parametric bootstrapping method, this is one way to produce the disruption in the throughout the random replacement of the site. So we'll be considering the sequence, we'll be randomly replacing the particular alphabet from the sequence, and then we'll take the statistical data, and that way if it is significant, like if it is obeying or it is rejecting the null hypothesis, that activity is going on. And second one is the parametric bootstrapping. Here entire sequences particular logic we will follow, will be changing that parameter, and finally will tally with the statistical significant way whether it is rejecting null hypothesis or accepting null hypothesis. Based on that, we can tell that alignment and its related tree whether it is correct or not. So in non-parametric bootstrapping method, it is what we are following. Suppose we are getting as per the tree and as per the normal activity of the software, we got this kind of alignment, and this is the related tree. So 1, 2, 3, 4, 5, 6, 8, these are the different alphabets are there. And if you look at the first position number 1, so first according to this alignment, so the position number 1, suppose I may here haphazardly need to change by following a sequence. So suppose during the bootstrapping activity, what the software or statistical informations before coming, what actual software will do. So it will try to take the first sequence entire thing. It is going to replace the sequence number 2, and that way once again it is going to extrapolate the informations and try to create the phylogenetical tree. Like that, second position, if it is written as such, suppose third position is going to replace by the entire second row column, whatever the informations are there. That so 1 is indicating first, second column we have replaced, the software will be replacing, and it will be placing the first alphabet, order the alphabets are there in the first line. So it is going to place there, and that way again it is going to get the information creating the tree. So like that, it will be changing with all particular alphabets, whatever is there. Suppose you have provided 100 different alphabet in one particular line of sequence. So with all respective 100 positions, it is going to compare, it is going to change, and once it is changing, every time it is going to create the this kind of phylogenetical tree. So bootstrapping, where this is indicating how we are tapping in with the help of boot. Suppose we are using a boot and we are putting step one after another. That means one place to another place. So that is the meaning of bootstrapping. So 100 different positions randomly it will be selecting, and it will be creating the tree. And out of creating 100 different bootstrapping method, whatever the tree is prediction is coming maximum time, so that tree information actually going to take, and software is going to present or depicted that particular tree. So that is called the bootstrapping tree, and this tree is going to make the consensus tree, and this tree is actual tree, and this tree whatever the information is going to give, that will be the 100% accurate and the correct. So this is the software is just doing the permutation combinations, and we this way it is working. But the actual statistics of it is like we can tell this is the Bayesian simulations or the Jacklifying statistical method, otherwise Kixino-Hagawa testing method. So here the standard deviation and the standard error both is going to consider. And as I mentioned that your null hypothesis is going to accept or reject. So here the statistical rejection value, it is a p is less than 0.05. So if the standard deviation and the respective value, if it is coming less than 0.05, then that tree topology is completely going to reject. Otherwise, the tree should be accepted. And that way, how many maximum rejection we are getting or maximum acceptation we are getting, that we are considering. Rejection one we are completely discarding, and software the maximum accepting part. Again, they are trying to find out the correlation between them, and correlation regression again analysis is going on, and finally the tree will be presenting like this way in front of you, and you will get the complete idea about the tree, and that would be your 100% accurate tree. So this is the all about your phylogenetical tree and how to create the phylogenetical tree, what kind of information you are going to get from the tree, and why this tree is very important, and at the same time why the phylogenetical tree also is very important for us to understand in the bioinformatical field. Thank you. If you are having any doubts, please you can ask me questions. Thank you all.
Participants,
Hello.
please have the interaction session.
Hello. May I?
Yeah, I can hear.
Yes. Okay. What is the difference between the guide tree and phylogenetic tree?
Yeah, guide tree only just haphazardly whatever the sequence we are providing. Suppose I have given my hemoglobin sequence first, then I have put it another hemoglobin sequence say for chimpanzee second, then I have given the sequence say for example from rabbit third. So automatically 1, 2, 3, 4, how many sequences we are given? 1 and 2 software is considering this is the one group, 3 and 4 next group, 5 and 6 next group. So they are not analyzing actual statistical information or whatever, fine. So this is called a guide tree. So whatever initially they are considering, now among them they are calculating R value, R dash value, or all kind of parameter, and then predicting the actual phylogenetical tree. So that is the difference between the original guide tree and actual phylogenetical tree. And in the guide tree, all the length, branch length, everything will be the equal. Whereas the actual phylogenetical tree, we can get the branch length, distance is completely different, everything. That is the difference. I hope it is clear.
Yeah.
Thank you. Anything else? Sir, in chat box, anybody has written anything? Just kindly. Is any question is there in the chat box?
I'm seeing. No one has.
Okay. I think then no one is having.
I have another question.
Yes, yes, please.
There are so many platforms are present
Hmm.
for multiple sequence alignment like T-Coffee, Maso,
Hmm.
Mafat,
Hmm.
and Cluster Omega.
Hmm.
Is there any criteria to select anyone?
See, there is no proper guidance to selecting which supposed to you are going to use. As I mentioned already, these are all whatever the name you have any means correction criteria on your hand. Whatever they have created, they have fixed the criteria, fixed the algorithm. You have to believe that, and you have to execute your result. But as I mentioned, the Mega, these are kind of software. It is well known and very famous one. So if you're using the Mega and if you're creating the phylogenetical tree and their bootstrapping method, neighbor joining method, what exactly you are looking for. So it is there in your hand. You can go ahead with that and you can get your result perfectly.
Okay.
But for tea coffee, basically that is going to give you a rooted tree information, no doubt. But problem is, that's how I mentioned, if you are looking for neighbor joining method to construct the tree, if they are having UPGMA method, so you have to depending on UPGMA method, not the neighbor joining method. So if you're using always, it is there in the bioinformatics always, if you're using any open resource, so at least 4 to 5 different open resource you use with the same data and you try to tally whether maximum of each is giving the same result or not. That is the only way.
Sujit Kumar Behra is raising. Sir, yes, come on.
Yeah.
Sujit Kumar Behra, you have raised your hand. Dr. Sujit Kumar.
Please, you can ask.
I put it in chat box. Means I hope the mega will be the best. I think, sir.
Yeah, Mega will be the best one. Mega always will be the best one.
So I think through mega the multiple sequence alignment by approaching cluster W, then we can make this phylogenetic tree with all bootstrapping, everything can be possible, right, sir?
Yes, yes, exactly, exactly, sir. Mega is always best.
So what will be the means cost of this mega software approximate? Because as you
no, it is cost exactly. I cannot tell you because I do not know actually. So you have to get it through institution and institution log ID and many things are required. And based on that, you can.
So we have to subscribe through institution, right, sir?
And institution wise, suppose 10 days or 15 days, they'll be giving like student learning one, what is called certificate. Okay. So you need to put the certificate ID, everything. The password actually they'll provide.
Okay.
And with the password only you need to access and you need to download and many one after another one on things will come. You need to go solve.
So in yesterday's class, we looked into the blast and FASTA. So any doubts with anyone related to the blast or FASTA, whatever we performed, specifically with reference to gap opening, extension penalties, or e-value, or identity, positives, PAM matrix, Plasma matrix, right? The values associated, PAM1, PAM250, like that. So if there are no doubts, we will continue with the pairwise and multiple sequence alignment, and we'll see if time permits, we will do with the phylogenetic analysis also today. So when my first question, okay, if someone can make an attempt, so or I will just show you. We did with the blast yesterday, and when we performed the blast, we obtained the alignment also. Now, when we have obtained the alignment, right, if you can recall, let me just check if it 51681, if it is going to take the sequence. Okay, it has taken. Then we made it like uniprot swissprot, and then we did the blast, then we obtained the result. Now, when we are having an alignment obtained in the blast result itself, why should we do a pairwise sequence alignment? Okay, so if I have to repeat the question, okay, let's say this one, right? This is with CCR5, maybe with this chimpanzee, right? We have an alignment present here and showing like 99% identity or 100%. If we take something else as an example, right? When we are getting the alignment here already from the blast, why should we conduct again a pairwise sequence alignment? Multiple sequence alignment, fine, that's a different concept altogether because we want to compare. But why should we do a pairwise sequence alignment again? Any guess?
Multiple sequence alignment is extension of pairwise alignment.
Okay.
So in order to cover all the sequences, so it has to be hierarchically aligned pairwise so that it covers all the sequences. It's a hierarchical extension of pairwise alignment.
Perfect. And now to go specifically with the pairwise alignment, okay. Can you tell me what is the difference between the local alignment and a global alignment?
Global alignment tries to cover the entire length of the sequence starting from the first residue to the end of the residue, whereas the local alignment looks at the identity. So only the identical sequences are aligned first, and then later the other sequences along with the gap they are aligned.
True. Perfect. And now the next criteria is when should we go for a local alignment and when should we go for a global alignment?
Global alignment if the sequence length is diverse. So usually, if the sequence length is of same length, then we usually go with the global alignment. If the length of the sequences are diverse, if the sequences are diverse, then we go with the local alignment.
Perfect, sir. Perfect. True. So that's the situation when we go for the local or global alignment. Okay. So that's the, I hope the understanding, whatever Dr. Mahesh sir has given, if all has understood the concept in the same way, then it's a perfect understanding. Global alignment or global sequence alignment we do when we have a similar over their entire length. Okay, if there is a difference, okay, if does not assume that the two sequences in question have similar over the entire length, then we go for the local sequence alignment. So if similar, okay, not the exact, so we may not have like a exact sequence length. If it is like 350 and another sequence having like 370, 380, 395, still we consider to be similar length. So we can go for performing the global sequence alignment. But if the sequence length is varying a lot, maybe like 352 with 400 or 500 amino acids, then we prefer to go with the local alignment. And we will look into what is the difference, and again we will work out with understanding about these gaps and other steps, okay, today. Now to understand this local, even in the previous talk from the Diptendu Sarkar, so there are two algorithms that we widely use whenever we do with the local or global alignment. By any chance you can tell me the algorithm names? if you can remember, in the presentation, professor has explained or mentioned. So we have like a dynamic programming, okay, that we use to perform this alignment. And within this, we have two types of algorithms that we widely use. One is called Needleman and Wunsch algorithm. This we use whenever we do with the global alignment. We use Smith-Waterman algorithm, this one, Smith-Waterman algorithm, whenever we perform the local alignment. The concept of working, okay, how it identifies the best optimal alignment is almost the same whether we use with the Needleman-Wunsch or Smith-Waterman algorithm. Only the scoring method is going to vary. So since I'm not going into the theoretical aspects, okay, let's start working with this. So Needle is one such program that we use for the global alignment. Water is another program that we use for local sequence alignment. Now to perform this global alignment, global pairwise sequence alignment, we need minimum 2 sequences. Okay, so let's click this needle and then it is going to ask like 2 input sequences. Now since we are working on the global sequence alignment, okay, now it is better to know what 2 sequences we need to take. Now as I mentioned in the beginning, first sequence remains human CCR5, okay, that we will fix with. Now the question is second sequence, which one to take. Now first one we will go to UniProt SwissProt, okay, and then here we can, now you should be able to type like P51681, that's the accession number, or you can type human CCR5, both is fine. Then our first sequence, why the search is not happening. Okay, so to get the FASTA sequence, okay, you don't need to go inside this and then look into the sequence. This is fine at the beginning level, but once you are familiar with this, so even if you have this, you can just select the accession number and then go for download, and then say we have selected one, then say go. Okay, we have the FASTA sequence available ready-made. Okay, so that's the easiest step. If you want me to do it again, select this, whatever the accession we want, then go for download. Then it will ask 2 options, whatever it has shown, whether you want all 88,000. If you select this, it is going to take, we are not interested in comparing all 88,000. So we want one sequence. So say download selected, only one. Keep everything as it is. If you want to download the sequence, then you say like compressed. If it is not the case, you can just say uncompressed, then say go. Then we have the sequence. Copy entirely, including the greater than symbol. Copy this and then go to needle. Keep your first sequence human CCR5. Now the second sequence is what is the interest. Now since we are doing with the global sequence alignment, we need to take a far related sequence, okay, because global is, we want to check which region is conserved, which region is not conserved, right, where is the dissimilarity and where is the similarity or conservedness, all those things. Now in such situation, okay, what we can do is we can take the sequence when we performed the blast yesterday, right, we got to know like the 50th hit, or the far related sequence, which is of a rat, right? So here it shows like it has 55% identity, and the first from the 9th residue of our sequence till the 352 positions, and there are 3 gaps present. There is 1 gap present here, right? So though it is going to give the result, but why should we go for a global sequence alignment? Is there any possibility the first 8 residues that are missing here, is it like at least one residue is going to match with this subject sequence, or is it like none of the residues is going to match in the first 9 positions or first 8 positions, because this is the 9th position? So when you want to address such conditions or questions, then the global sequence alignment becomes an important tool. Since here we are getting a closely related, in few cases, right, the sequence alignment itself will start from, let's say, 50th position or 20. Now here it shows like in the 20th position it is starting. Now if somebody asked like, okay, your first 19 residues of the human CCR5 is not matching with the CCR3, right, CCR3 protein, that is fine from the blast result. Did you check at least 1 or 2 residues will match out of this 20 with the CCR3? That we won't get from the blast, because the first 20 it is not considering. Why it is not taking first 20? Because the threshold value is not going to match if we include them. So that is why it has not considered. Now can I do pairwise sequence alignment manually and see whether some residues is going to match or not? At least 2 residues will match or not. Then we will go for manual method, that is this. Now we can take this CCR3. Now the interest is whether CCR3 and CCR5 are related to each other or not. Okay, but the organism is different. If we are interested, that's why I am mentioning based on your question, okay, what you want to address. So we can also give a criteria like Homo sapiens, only include Homo sapiens. Then we can perform the blast. If my interest is how is the other CCR proteins, CCR1, CCR2 or CCR3 is linked with CCR5, then we will give the criteria like only human, and then do the global sequence alignment to check how is the variation between them. So if my interest is, let's go to the alignment. Okay, now this is with human CCR2, this is with human CCR1, everything is from Homo sapiens, because our criteria itself is to take only Homo sapiens. Then CCR4, then CCR3, right? So CCR6, right? CCR7, then we have like other CXCR4, right? Now we also have like CCR10. Okay, now looking into this, we feel like there are many gaps. 5 gaps are introduced in the local alignment. Now the number of gaps will increase when we do the global alignment, or it will decrease? When we do the global sequence alignment here, by taking this, for example, if I take this protein CCR10, whether the number of gaps introduction in this when we perform the global alignment will increase or decrease?
Decrease.
Yes,
Is
increase.
because we have to align the sequence throughout the stretch.
Perfect. So to cover entire stretch, so it is going to include the gaps to cover because the aim of introducing the gaps is to cover as many residues as possible. Now to do that, this is the accession number. Let's take the CCR10 as an example and then perform the global sequence, pairwise sequence alignment. You can right click and say like open in new window. Then we have the sequence. This is having 384 amino acids. Our CCR5 is having 352 amino acids. Since we are interested to see where and all it is conserved, right? So you can say like the FASTA here. So the sequence remains the same, okay, even if we take from, because they have already given the information here. So this is the accession number from the UniProt SwissProt. So even you can copy this and go to UniProt SwissProt and type and search, okay, and you will get the same hit, or you can take, I don't know, I should use another browser maybe. Otherwise you can still take this sequence and then do either of the things is one and the same. Now you copy the entire sequence, copy this, and then go to pairwise and paste your second sequence. Okay. Now one of the best practices is, okay, because when we do with the multiple sequence alignment and other aspects, if the description line is like this, okay, it becomes too difficult to work. So the description is for our understanding. So when we know what it is or other aspects, you can remove it off. So you can just remove this, keep only human CCR5, rest all the things you can remove. Okay. Here also, this is coding for CCR10. So you can just delete this. Let us say it is given like ACKR2 human, right? So other lines you can remove. But make sure your second line starts with the sequence, okay? Don't make it like this. Then this also becomes like a comment or description. So the second line should start with the sequence. First line after the greater than symbol, it should be just with a few letters, okay, so that we can easily understand. So now CCR5 we are comparing with ACKR2, okay, two sequences having a varying length but not too much of a variation, 152 and 180. And how the output should be, keep it as a pair, okay, default. So we will also go with this file format or cluster format, other when we do with the pairwise or multiple sequence alignment, keep this one pair. Now we have more options here. Again, if you increase or decrease the gap penalty, you have an option. Gap extension penalty, you have an option. How the alignment score should be calculated, you have an option to select the matrix, right? If you look into, we have so many varieties like 30, 35, 40, 45, 50, right? Blossom 90 up to that one. Then if you want to see the PAM 10, PAM 250, PAM 500, like that. So let's keep the default, everything as it is. So end gap, if it has to put some gaps at the end, how should be the penalty? Those referred as end gap penalty. So let's keep everything default and then say submit. When you are submitting too many sequences, then we go for this notified by email, then we can provide. When we are having like two sequences, if the length is too much or if it is taking long time, then we will use this, otherwise it should be fine. Then say submit. Now what we should do is, we have an alignment here. It shows like in the blast result, this is also a pairwise sequence alignment, but it's a local alignment. That's the only difference. first 19 residues is not matching with the first 35 residues of the subject sequence. That is what it is showing. First 19 is not matching with the first 35 of the subject sequence. Now, even in the human CCR5, it is going to end at 311 itself, but we have 352 amino acids. And here also it is ending with 326, but we know the length is 380 amino acids for the CCR10 or ECR2, either of the things, a typical chemokine receptor 2 or ACKR2, either of the things. Now, now this is done. Okay, now the pairwise sequence alignment is done. It is recommended that you save this particular page. Okay, when you are doing, okay, save this particular page, because when you are writing a paper, you are supposed to write these also. Gap open penalty, what was the penalty that you have put, what was the matrix that you have used, okay, and the end gap penalty, what is that you have given. So these things you may need to put, so that if somebody wants to repeat the experiment, they will also get the same output. Now, if we look into this, right, there are 70 gaps introduced, and the percentage of identity is only 32%, and percentage of similarity is 48%. Now, if you want me to keep it on a side by side for your comparison, we will keep this. We'll keep this. Okay. Now, the advantage, why should we do this global sequence alignment is, so it is given the information that first 19 is not matching. So the first 19 comes somewhere. This is the first amino acid, MDY. But D and D is conserved. These two amino acids are matching. What it is starting is with the CRKDA. So this CRKDA starts from here. So that is the same thing like CQK, right? CQK. Now in the BLAST, it has not shown this particular alignment at all till this particular region, right? The alignment it has not shown. Why? Because there are many gaps. When the gaps are there, there is a penalty, and when we have a penalty, the score will get decreased, right? Score will be less. Now the percentage of identity, if we look into here, right, it shows like 38% identity, but here it shows like 32% identity. Okay, that is where you got to be clear, you got to check whether the alignment is a global alignment or a local alignment. We always feel, maybe I will just decrease, 35% identity is greater than 32.3% identity, right? But the coverage is how much? And that is where the query coverage comes into picture. But it's a local alignment. So within the local alignment, we have 35%. But in the global alignment, covering entire sequence, we are having like 32%. Okay. So this is important. If somebody asks like, is there any amino acid which is going to match with the first 20 amino acids of the ACKR2? Why I renamed in the beginning is, we can easily see the names here. If it is not given like this, it will just show like SP underscore something. So which becomes too difficult to see to which particular protein it is coding for. Okay, that's the reason we will edit the first format. Now we know that there are 1, 2, 3, 4, 5, 6, 7 residues are identical, but the BLAST did not consider because these gaps are present. Again, why the gaps or why it is not considering? Because maybe it is an insertion. These residues may be an insertion. Okay. Then the other portions will continue. Again, if you look into, there are some gaps introduced. Yes, whether the gaps are introduced here also. So overall picture we are going to get, okay, whenever we perform the global sequence alignment. Now here it shows like after this VGL, that's the 352 position, nothing is matching. That is why the gaps are introduced in our sequence, human CCR5, because the length is less here. So the length in our sequence is less. That is why the gaps are introduced in our sequence. Here also. Now further, if somebody wants to go for a critical analysis, okay, because why should we do this analysis, right? What is the purpose of doing this? That is the question, right? Now we need to take this structure. Finally, in all the aspects, whatever we do, the hierarchy goes like sequence to structure, structure to function. Okay, in most of the cases we know the function, but we do not know its structure, because structure determination is a complicated process. But we have the sequence information. Are you getting? We have a sequence information. We do not have a structural information for all the proteins, but more or less we know the functionality of those proteins. Now, if that is the situation, what is the way by which we can build the bridge between the sequence to the function if the structure is not available? So that can be done through this pairwise or multiple sequence alignment. Now, when the sequence length is varying between the human CCR5 and other protein, what makes these two proteins to come still under the GPCR family? Okay, or how many helices are there even in this? We will do all those things in the later point of time also. Okay, pro-site signature analysis is there. At that time also I will ask the same question, like if you got to recall these experiments, whatever we done. Now if we look into, right, so this region is highly conserved, this region is highly conserved, and this region is highly conserved. So if you want, okay, you can just spend another 5 minutes of your time. We know already the human CCR5 topology, right? Whether the helical region, whatever, I think I will put this one here. Okay. Now, whatever the helical region that is there in the CCR5, whether it is conserved or not, that's what we can check now, because helix, beta sheets, and trans are the secondary component elements. Now, the helical region starts from 31 to 58. In this 31 to 58, is it conserved? So 31 is this one. 31 to 58, somewhere around here, right? Yes, the sequences are conserved between these two. There are many identical positions. Then the second helix, 69 to 89. If you just check again, 69 somewhere around it starts, and 89 means somewhere around here. This is how the 81 is with the human CCR5 position. Okay, so the counting will happen only for the positions, not for the gaps. Okay, so there are gaps introduced, but the 31 refers to we have 31 amino acids present. Here we have 60 amino acids present continuously. So each length, okay, now that is where you have like 81. Then again continuous 131, it will add. So the gaps, if it is there, that is not going to add. So like this, if you check, without performing the structure prediction, right, we know the structure of the human CCR5 now. But without going for the secondary structure prediction of this ACKR2 human, we can easily guess like how many helices this protein may also have, because the first few residues are not matching. So last few residues are not matching. They are the end, C-terminal and N-terminal. Now the helical regions are reaching, or helical regions. First helical region is matching, second helical region are matching. There are some gaps. Whether the gaps are there in the helical region or non-helical region, then the third, fourth, fifth, sixth, seventh. So when you check like this, you can easily estimate or guess whether the ACKR2 is also going to have the 7 helices or 5 helices, or how it will be. Because you have direct information here, you can easily correlate and link it up. If the helical region is this, and if it is conserved, then in the ACKR2 also it is going to code for the helix. Okay. If in your protein, if we take another protein, if it is a beta sheet forming residue, stretch is coding for a beta sheet, for example, and in this ACKR2 also, if it is conserved, then you can easily guess that that is also going to code for a beta sheet. And motif, if it is there for the, that we will do motif analysis. So if there is a motif present in the CCR5, and if the same motif, let us say if this is the motif for the CCR5, and if it is the same motif that is present in the ACKR2, that means there is a high level of conservedness between these two proteins. Or if we go for a domain, and if the same domain information is present for this, then also we will get to know like, yes, that is also going to do the similar functionality. Okay, so that is how the extrapolation will be made, or that's how the correlation will be built between these two. Now, can we convert this global alignment, whatever we have, into again a local alignment? In other words, I don't want to see these end gaps, because I know that it is not coding or not aligning with this ACKR2. So is it possible that these gaps should not be introduced and then get only starting from this ND? Okay, MDY, whatever is there, and ADS, whatever is there, from here it should show. So the first few not to be there, it's not required to be represented. Then what should we do? What we can do here? Just increase the end gap opening penalty and end gap extension penalty. Okay, then just check. So when you increase, it is going to work only at the ends. So those end gaps, it's an iterative process. Okay, so if we still get those end gaps, that means we will get to know like, yes, there is a still need to increase the penalty. Okay, still if we are getting this, so the penalty whatever we have assigned is not enough. So again we get to come back and then again increase this. Okay, then those gaps will be removed off. So it's all statistically related, what type of amino acid we have, and what kind of changes to be made. Okay, so it works very systematically. Okay, because the conservedness it looks for, threshold finally it looks for. Now the entire penalty itself is increased. Now the number of gaps got decreased from 70 to 34, because the gap penalty is increased very much. Accordingly, the identities also get decreased, similarities, right? Number of gaps decreased. So we cannot see any gaps because the penalty is so high that no gaps are introduced in between. But there are too many mismatches now. Okay, so if we want it to spread, decrease, again decrease the penalty. So the purpose of performing the pairwise alignment is to check two individuals entirely. Okay, now it is increased. Gaps are introduced here. Even if there is some mismatch, it's something like this. I have 5 fingers here and another guy is going to come with only 2 fingers, and then we want to check this. Local alignment is something like, okay, just check this, and this, yes, this is where it is matching. Then if it is continuous, then it is going to show like 2 fingers are matching, and then it is going to show the results. But if there are 2 fingers, something like this, which are, and if you want to see where it is matching somehow, then you will go for a global alignment. It will show this finger is matching and it will show this finger is matching. In between, it is going to put gaps. Then you will get an idea, okay, this finger and this finger is matching. The middle 2 finger information is lost. Okay, so these 2 fingers are matching the middle information. But in one hand, out of 5 fingers, entire information it is going to show. But in other sequence, whatever the 2 regions that are matching, or let us say the 2 regions may be these 2 or these 2. Only these 2 information it is going to show. In between, it is going to put a gap. Then we will get a feeling like, okay, the 4 finger and the little finger information is what is available. So the other finger information is put as a gap. That means those finger information is not there, or those fingers are not there in this particular organism. Okay, that is what we can. But when we do the local alignment, it is going to show only the thing which is going to match this finger information and then this finger information. The middle one it will skip. It is not going to introduce the gaps. So to know where they are present, how big is that, how big is this, all 5 fingers, where it is matching, to get that we really need to do the global alignment. Is it clear with this concept? Yeah. Now for the same sequences, okay, let us do the local alignment and check how it looks like. So select the local. Now the Smith-Waterman algorithm, provide the same human CCR5 as your first sequence and provide the same second sequence here. Okay, and then again we don't need to change any parameters. Let's keep everything default and then say submit. Now if we look into, right, this is going to be the similar result whatever we obtained. Where did it go? Last result. Okay, this is the one of CCR. What was the accession? ACKR2. Now this result, this is also, now this is going to be the similar like whatever we are going to obtain from the blast. So first 20 is not matching, then comes with the 36th position, and then it shows. Now if we want to do further refinement in this, okay, let us say it has introduced 9 gaps, okay, to make it a optimal alignment. I do not want any gaps to be present. Okay, it goes in a very specific manner. The 2 fingers, this is where it is matching. Okay, and further question is, within these two fingers that are matching, okay, is there a way by which I can see which core region is highly conserved? Okay, which core region is highly conserved? There are so many similarities that are there, right? And there are some gaps introduced here or some gaps introduced here. I do not want even a single gap to be there. I want to see a highly conserved region between these two, highly conserved. Then go back to this, okay, increase the gap penalty, increase the gap extension penalty, and increase the blossom to 90, okay, because I want to see the highly conserved region. Don't introduce any gaps, don't introduce, if it includes, don't extend that, and I want to see the highly conserved region. Then you put this one and submit. Then if you look into the gaps introduced in the alignment will be reduced. Previously it was 9, now it is reduced to 1. Okay, there is one gap that needs to be introduced inevitably. Okay, so I can just zoom in a bit. And that gap will not be usually at the end. Okay, where is the gap? Yeah, here it is. So there is only one gap. If this gap is not introduced in this protein, the alignment is not going to happen at all. Okay, but we will get to know like the regions which are highly conserved. Now the gaps are removed, the position shrinking has happened, then we know that this is a highly conserved region. Okay, that means this region has not become like aligned by chance. Okay, it is really highly conserved. That we will get to know when we increase the gap penalties or select a higher blossom value. This region becomes highly conserved. This is another region, right? Highly conserved from here to here. There are many places where the changes have happened. Because of these changes, maybe the protein is doing a different functionality, okay, but still belongs to the same family. Different functions, but belongs to the same family. Okay, so that is how our interpretation goes. Any doubts with this? When should we go for a pairwise, and within that, when should we go for a local alignment and global alignment? This is important. Okay, even if you want, I can just show with one of the paper. Okay, when we worked out, that is when it's a part of my PhD work. So where is the mutation happening, and how the two proteins are getting changed? Okay, all those things we can understand by doing this multiple. When we do it the multiple, I will explain, but pairwise is also in the same concept. Okay, so I have 2 from the mammals, 2 from the insects, Bos taurus and Homo sapiens representing the mammals for the actin protein, Drosophila and Helicarpa representing the insects. Then we look into where the mutations are. So we have methionine and methionine in the mammals, leucine in the insects, threonine and serine, phenylalanine and tyrosine, serine in the Bos taurus or Homo sapiens, and alanine. Now, what makes the intention of this particular work, okay, is you may be knowing that phenolurate is one of the synthetic pyrethroid which is used as a insecticide, pesticide. It has a high affinity for the insects, but still has a low mammalian binding affinity. So what makes this phenolate to have this binding affinity? So it's a potent insecticide, moderate toxicity towards mammalian beta-actin. It's also there. So why it makes this to have a moderate toxicity? Because it has to work only with the insect. Why it is still acting with the mammalian? But why, when we have the beta-actin present in both insects and mammals, right? Why we still have a moderate activity? Why not the same way as that it is going to affect the insects, right? So then the purpose was to check at the sequence level. So we did this is the structure of the phenolate having alcohol moiety, central ester and acidic. Then perform the multiple sequence alignment, then get the structure, then perform the molecular docking, then see how is the interaction. Okay, so when we are working with the molecular docking, I will elaborate on how to design your experiment and other stuff. I think I also have a table. Okay, if it is not there, it's gone to. So we will get to know like on what basis it is going to have the mammalian acting, how to interpret these results. These things I will explain it out when we are working with the molecular docking. Okay, so multiple sequence alignment or pairwise sequence alignment becomes a useful tool to know where and all the variation is there. And as I mentioned, we already know the function. Okay, coming back to the principle or the core, we already know the functionality, that is having a moderate toxicity, but we do not have a ways to prove or understand why it is having. Whether the structure is there, structure is not there. Whether the sequence is there, yes, sequence information is there. So take the sequence information, do the multiple sequence alignment, get to know where and all the changes are there. That is the sequence level. Next is take the structures and work at the structural level, understand at the structural level why the variation in the binding of this phenolate with the mammalian actin and with the insect actin. Because of that difference of the interaction, it is having this moderate activity. That is how we are going to make the conclusion. So to do that design, it is better to know all these principles or the basic concepts. Then it helps us to decide whether I should generate a local alignment here, or do I need to generate the global alignment, right? So you can make out those decisions. So where it starts, how many amino acids do I need to include? Do I need to skip anything? Is there any gap present? Do I need to keep those gaps? How many base pairs are there in Bos torus? How many base pairs or residues are there in the Homo sapiens? Is the length same in all four organisms? It is a beta-actin, but whether the length is same or the length is different. So if the length is same, which region is highly conserved, right? So if you look into, right, all the amino acids are highly conserved. Star, star, star. This star represents highly conserved. Colon is semi-conserved. If we get like a dot, that represents the partially conserved. Now here it shows like it is a mismatch, right? There is no symbol, but this is semi-conserved, semi-conserved. So even if it is a semi-conserved, whether it is still having that effect, right? Where the ligand is interacting, whether the ligand is interacting with these amino acids where the changes are happening. That's how you will get the complete holistic view of your work. Then you will better understand. So to generate this is what we are working out now. Okay, this is one such application why we do this pairwise or multiple sequence alignment. Now let's move to the next one, that is with reference to multiple sequence alignment. Okay, now in EMBL EBI, okay, just as additional information, if you go to the services, okay, there are many tools, many databases available, and different tools available for performing different activities. Okay, it is just you should know. If we are working at the DNA or RNA, if you click right, the services that are available for the DNA or RNA that it is going to show. We can use this clustyle omega to do this multiple sequence alignment. Blast search we can do, right? We can do with the genome analysis, ENA sequence search, right? Enzymal analysis, and all this is at the level of nucleotide, either with RNA or DNA. If we want with the gene expression, there are different servers available. Protein, we have different servers available. So my suggestion is you don't need to remember these things. Okay, even I will repeat. If you go for remembering or remembering, basically by hearting or remembering, right, it becomes too difficult. So the way I am looking at this particular workshop is, you're all new participants. Like, let's say if I am new to a city, right, if I am visiting to Chennai, let's say if I am a very new guy and visiting to Chennai for the first time, my aim will not be to remember all the names, in the sense like what is the petrol bunk name is, what is the name of the theater, what is the name of that, this. No, my intention will be to understand the basic stuff, something like where is the bus stand, where is the railway station. Later on we will get familiar with knowing what is the name of the railway station, okay, what name they have given it. But my first purpose is just to know where the railway station is, where the airport is, or where the bus stop is, where the hotels are present, where, in which area I should stay, where is my working location, how far is that, right? So as an overview we will be looking at. Then we get used to saying like the bus number or your station name or all those things. You automatically, you will by heart or you will get to know. So in that particular context, okay, so in that particular context, it is better you remember that there is EMBL. Okay, that's enough. And based on the purpose, whatever you want, you can use these services. Okay, because if you go for remembering all these things, you may miss out the big picture. Okay, so you should not miss out the big picture. What is that possibilities you have to do? Okay, that's what my intention is. These are the possibilities. It is just as I mentioned, you should know the shops available. It is not like you should go to those shops and purchase anything. If you know, when time comes, you know that where you should go. That's the only thing. Now in that particular context, okay, let's go to this protein. We want to do with the multiple sequence alignment. So just click on this. And also there are tools which are standalone, but whenever we are doing the proof of concept, or whenever we are just checking, right, whether the thing is going to work or not, so you don't need to do it in a very systematic manner. So you just want to check whether it is going to work or not. So this is going into web API. I think sometimes if we go with this, that becomes too difficult. Okay. Now, how many input pages will be there for multiple sequence alignments? We have 2 for the pairwise. For multiple sequence alignment,
At least 3. At least 3.
at least 3. So 3 sequences are required. Minimum 3 sequences are required to call it as a multiple sequence alignment. The input page will have only one box, okay, because the tool does not know how many sequences you are going to give. Someone may give 100 or 500 or 1000 or 3000 sequences also. We have performed the multiple sequence alignment. So that is why we have only one box, and we can provide the sequence over here. Now, from where we will get the multiple sequences? We need sequences to do this.
From blast search,
From the blast,
there will be multiple arrangement of sequences. We can select our required sequences, then we can save it in a notepad file and then copy paste.
perfect, true. So we can go back to our blast result, whatever we have, and then we can take the accession numbers and then do the multiple sequence alignment. Okay, can go for the descriptions, and then we can select the sequences of our interest, something like this, whatever we are interested. Or another way is, this is one way, and another way is if you just type like, if you say CCR5 rather than giving like human CCR5, okay, you here also you will get to know like CCR5 of the human is there, CCR5 of the mouse is there, CCR5 of the rat, CCR5 of the dog, chimpanzee, gorilla, bovine, and then pongo, rhinus, right? So rabbit. Now you have the list of animals and the CCR5 that is being present. That means the protein is present in all these organisms. Okay, that is one way by which you can take the protein sequences. Or another way is also like when we go to GenBank, I'm just telling the different possible ways, because when you are working you can pick any one of it. Is not like there is a standard procedure, okay, because for human CCR5 we have this kind of a data, okay, but for your protein of your interest there may not be RefSec available. Then again you go back to this, rather than taking from the Swiss prot you may take it from the Tremble, okay, or you may select from the, sorry, you may select like this also, but nothing wrong, okay. The way is different, like you can pick the sequences from different sources, but the why are you doing that, that is most important, okay. The purpose is most important than the execution, okay. So either you can take like this or you can go to this RefSec, okay. Then we have like a orthologs information given here, so you can select this. There also we have 66 sequences. This is from human, monkey, chimpanzee, sheep, right, and another organism, chicken, goat, rabbit, olive, baboon, right, western gorilla, horse, bacterium, domestic ferret. Now if I do not want like this, right, there are too many organisms, then you have option here to select. If I am interested in birds, you select this and there is only one. But if you want with the mammals, select this. So we have 65. Now within this mammals, I want to go further refinement. I want to take the organisms which are carnivores, okay. Then you have an option to select only the carnivores or only with the primates including humans, so you can just select the primates. So you have these organisms representing the primates, that is around 29 sequences, okay. So based on the, as I mentioned, armadillo yesterday, right, so if you want you just select like this. So what, based on the purpose, one can select and then do. Let us say, let us take this primates, okay, then select all. So we have around 29 organisms selected. Human CCR5 is having 352 amino acids, and again other organisms, but all representing the CCR5, having a varying length. Okay, 352, 354, or 379, right? It's fine. .373 here, gray mouse. Now you can say download, and then say like download. Then you can save this file. Maybe I will save this one. Okay, and then it is better if you open this file. So we have the nucleotide sequence, okay, transcript variant information, but I want with the amino acid, right? So protein. So say protein, then download. Then save this particular file, the refsec. Okay, so we have the amino acid sequence for all. Okay, so the first thing is in your laptop or desktop, please install this Notepad++. Okay, don't use the Notepad. Install Notepad++ because this becomes useful when we are working with the docking also. Okay, in the next step. So just go to Google, okay, and then type here like Notepad++. Why we should not use Notepad is the formatting will not be maintained in the Notepad. So to avoid that formatting issue, I can just show you how it looks if it is with the PDB file or other files will not be in a formatted mode. Okay, if it is, if we open that in the Notepad, okay, so we prefer with the Notepad++. And also one more problem is whenever we go for saving the file in the Notepad, it will save it as .txt. So though you are saving it .fasta, right? We have it like .fasta, but it becomes like .fasta.txt. So the tool is going to give a message like not a right format. So it was not going to read that file. So to avoid all those confusions, better to install this Notepad++. Okay. Then we have all the sequences here. Control A, copy everything, copy, then go back to the multiple sequence alignment, plus tile W, let me close this. Okay, paste. Now if we run this, right, it becomes too difficult to know what exactly the organism is, but we know that all the sequences belongs to CCR5. So one can delete this and retain Homo sapiens for the first one, pan for the second one, right? You can do this even in the Word or Notepad, whatever I have it, right? Here also it becomes easy to delete. Sorry. Okay. So this is going to help to know the organism, you can retain only this XP, but again you have to go back and go for a mapping. What is this accession number is coding for? So I will just do for few, okay, but whenever we are working on the real project, so it is better if we remove everything. So if you can recall with the paper that I have showed, right? So the manual editing was made, so that is why you can easily make out that Bos torus, Homo sapiens, Drosophila or Helicarpa. Otherwise, we will have it like the accession number, so which becomes too difficult to know which organism it is coding for. Okay, so we will do for a few, as I mentioned. Let's keep other things as it is because it is going to take some more amount of time. And let's save this, then Control A. Okay, so make select all, copy, and then go back to our let's delete, paste. Okay, and the tool is going to work in such a way that if we have two entries, if this greater than symbol and the description, if it is the same, then we will have an error. Okay, saying like you have put two similar sequences. So the greater than symbol Homo sapiens, and nowhere the greater than symbol Homo sapiens should be there. Okay, if it is there, then we will get error. So that needs to be taken care. As we know, they all belongs to different organisms, right? It should be fine to work with. Now again, keep everything default. Keep these things are also default. Okay, no, you don't need to change anything, and then say submit. Okay, so this is a hidden Markov model iteration that it is going to use. Let it find, and how the order should be. So the input are aligned, whatever goes as a closest, that it should show first. Just submit this. Okay, now we obtained the result. Okay, if we want to give a coloring, we can give like show colors. There is an option. So for each amino acid, it is going to provide the coloring. That's what even you can observe here. So the coloring is given for the amino acids. And then wherever there is a conservedness, we have a dot or a, I will zoom in. Okay, so this
Please
image,
repeat from where you have made
Yes,
the
sir.
coloring.
so you have an option here, show colors on the top. Hide colors or show colors. Show colors here.
Okay.
Yes. Now, all these proteins, whatever we have considered, right, this we know that it is from the orthologs. Same protein from different organisms is nothing but the orthologs. Okay, different proteins within the same organism is referred as paralogs. Okay, Homo sapiens, but CCR1, CCR2, CCR4, CCR5, if we take within Homo sapiens, within human, different chemokine proteins, then they are referred as paralogs. Same protein, CCR5, but from different organisms is referred as orthologs. Okay, so that is why it is a ortholog. Now, where is the result? Yeah. Now, if you look into this, for a few sequences, I have labeled Homo sapiens or Pan or Pongo, right, mouse. But for few we did not do. Now you can make out like we are only going to get the accession number. So now it becomes difficult to which particular organism actually this particular accession number is coding for. So we may need to have again one more lookup table, or we should have this here and then check to which particular accession number it is matching. Okay, so to avoid this, right, 230092, so 230092 wherever it is there, right, we got to check. Okay, then we will know like, okay, this belongs to this particular organism. To avoid that, it is better if we edit and keep only the organism name. Now, how to interpret this multiple sequence alignment? Now, when should we go for a multiple sequence alignment? Anyone? What is the purpose?
To align the more than two sequence as well as to relate among the organisms so that we can elucidate the phylogenetic relationships or evolutionary relationships,
True.
either if it is present or not.
Correct. Any other answers from anyone, from others?
To find conserved sequences among
Perfect.
them.
Yes, sir. To find the conserved sequences.
Yeah. To
Any
know
other-
whether the sequences are homologous to each other or not, to know the conserved sequences, and to infer the phylogeny.
Perfect. To get the phylogeny, to find the conservedness. Some more, if you can go for.
To know the conserved or the semi-conserved region, so to know the identity between the sequences, if they are closely related, if they are distantly related to each other.
Yes. So from the multiple sequence alignment, okay, so the first thing why we go for multiple is when we are not able to make a conclusion from the pairwise alignment. Okay, it's something like if I am going to say the time is 12:15 and if another person is going to say now it is 12:30, okay, that means we have a confusion. We do not know whether the 12:15 is correct or 12:30 is correct. So we have an ambiguity. So to remove this ambiguity, what we should do is we will ask a third person, what is the time now? So if the person says like now it is 12:15, then the first one is correct. So then the 12:30 is not the case. If the person says like 12:30, then it is 12:30 we should have. Or if somebody says like 12:20, right? Not 12:15 or 12:30, he says like 12:20. Then we have one more ambiguity between them. Then what we will do, we will go for the fourth one. Then we will ask that guy, what is the current time? If he says like 12:25, that's again ambiguity. Next guy, 12:29, that's again ambiguity. So you will keep taking the data to remove off the ambiguity. Okay, to remove the ambiguity. And then let us say if we have 10 members and each one telling different time. Okay, 12:15, 12:16, 12:19, 12:21, 12:23, 12:25, 12:30. Then we will have more confusion what is the exact time is. So what we will do is we will take some more data set, ask some more people what is the correct time. Now if somebody says like the exact time is 12:30, okay, then we know that yes, the time given 12:30 is the right one. Now the confusion, the core question is the moment you ask the time, right? So the time is also elapsing. You would have started at 12:15, but some other person said like 12:30, but you started taking the data, you started asking another person, maybe you took 2 minutes time to ask that question. So then the time of 12:17 is correct because 2 minutes is already lapsed. Then you have a confusion, you are taking. Then so you will apply your time, how much duration is already spent, and then you will take a decision, yes, this is what the correct time is. Now how should we apply the timeline, whatever the time spent, whenever we collect the sequence, right? So the timeline we will get from the whenever we perform the phylogenetic analysis, how much time, if I say like the Homo sapiens and then the span is closely related, but is there any other organism that has evolved in between? So to include that, right, we will take the time also into consideration. But one of the advantage with reference to the sequence is the mutation that it is going to happen in these organisms. Okay, whatever the organisms that we have considered, right? Sorry, primates, the mutation to be accepted by a natural selection process is a long time, usually in millions of years. So taking that into context and taking the molecular clock hypothesis into picture, we assume that the mutation that is going to happen in individuals is going to be constant. Okay, so that is why we are not worried too much about whether this sequence is correct or this sequence is correct, not like that. So we assume that all the organisms are surviving and all the organisms are healthy in nature, they are not towards the extinction. So whenever the evolution happened, we assume that the mutation has happened in all the organisms in the series of events only. Okay, that's the first assumptions we do in the phylogenetic analysis. Now, before we go for that phylogenetic analysis, the multiple sequence alignment has too many advantages. Okay, we use this multiple sequence alignment in almost all the aspects when we cannot make a conclusion just with the two sequences. We use this for the protein structure prediction, secondary protein structure prediction. We use it for the domain analysis. We use it for the motif analysis, right? We use it for family conservation. We use it for protein functional analysis, protein binding site prediction, active site prediction, right? Or the overall structural stability of the protein, the size of the protein analysis, for anything, right? Sequence to structure to function, the hierarchy, irrespective of the levels you are addressing a particular question, we use this multiple sequence alignment. So this is one of the again a basic aspect or basic step that we perform to make any conclusion. So this is just one such example I have given. Okay, so like this, whenever we want to do some kind of analysis where we need more than two sequences, multiple becomes a best option. Now in the morning it was asked like which tool to be used, right? Whether clustyle omega or clustyle W, mega, right? Emboss, within that there are. You can use any of the tools. In more or less, in most of the tools, the principle or the algorithm way that it works is almost the same. Okay, so you will get the similar kind of alignment even if you use any other servers. Okay, maybe when you change the gap opening or extension penalties or change the matrix, so these alignments may go for a slight variation or representation may vary. Okay, so we have it like star or a colon, but there they may put like M here or conserved residues, just like the whether the blast is better or FASTA is better. All right, so the alignment wise, in the blast we have the residues, but in the FASTA we have symbols. Otherwise the alignment looks or the interpretation remains the same. So when you are doing it the first time, okay, to get the hint. Yes sir, any doubts? Yeah, when you are doing it the first time and if you want to get some hint which organism is closely related or how is the relatedness between the organisms having the same CCR5, just do the clustyle omega. Okay, this is more than sufficient. We have more information available. Even you can use the, I would say this here itself, okay, in the UniProt itself, you can select the organisms, okay, and then you can perform the multiple sequence alignment. You can say align, okay, maybe I will use, I don't know why UniProt is not working fine with the Firefox.
Hello, sir.
Yeah,
I have one question that in multiple sequence alignment, what you have to do? There are some star markings.
yes.
Okay. So what is the meaning of that star marking?
Yeah, that I will do. The results, right? This one.
Yeah. In multiple sequence alignment.
Yes. No, we will spend some time in interpretation, okay, of this interpretation. So that's not a problem. I am just thought of showing you the ways by which you can perform the multiple sequence alignment. So interpretation more or less remains the same. So you can take the sequences and do it like this, or you can go to UniProt and then if you type CCR5, right, you have all these things. Then you select some organisms of your interest. Then if you say like align, then also the multiple sequence alignment will be done directly here, okay. So you don't need to go to like cluster omega. This is another feature from where you have the alignment done. So it makes life easy because you can easily pick, you have the protein name, CCR from the human, mouse, rat, right, that was already there. Then the interpretation now is something like we have a star represents identity, okay. In other words, we can also say like conservedness. Star represents conservedness, colon, I will zoom in, star is the conservedness, colon refers to semi-conservedness, okay, semi-conservedness, and dot, we will also have like a dot here, dot represents partial conservedness. There are three types that is there: conservedness, semi-conservedness, and partially conservedness. Now maybe someone can tell me the difference between these three now: semi-conserved, semi-conserved, and partially conserved.
Semi-conserved means same physico-chemical groups,
Yes.
and partial conserved means belonging to different physico-chemical groups.
Yeah. Almost correct. Not with a different one. Some more refinement.
Partial is not correct, I think.
Yeah. First one is identity. Okay. You can take this as identity. Now we have methionine present in the first column, right? So that means identity, 100% highly conserved. So you can also call it like highly conserved rather than just conserved, because the second one we say like semi-conserved. So highly conserved, semi-conserved. Semi-conserved because these two residues share the physicochemical properties much higher than whatever we have it here. If you can recall, right? So the tyrosine and phenylalanine belongs to the same group. It is, if you want to bring it back, hydrophobic and aromatic. If it is there, if both the properties are fulfilled, then they are referred as semi-conserved. but hydrophobic, but they are not the same. One is aromatic, another is aliphatic hydrophobic. Then we say it like partially conserved. For example, alanine and valine, right? So valine is aliphatic and hydrophobic, but not the alanine, it's just hydrophobic. Then we say it like partially conserved. Mismatch is they do not share the common physicochemical properties. They are different. Then there is referred as partially conserved. Now, if we want to put the blocks for this, okay, we can use these properties, amino acid properties here. Show me all the similar amino acids. If you take the similarity, now it is going to show with the color. The dark color shows highly conserved. The light color here, right, shows like partially conserved. And other regions, we can make out the three colors that are there. Dark background, highly conserved. Then the lighter one here is the partially conserved or semi-conserved. Still lighter background is partially conserved. The regions where there is no mismatch, it is kept. No background is given for this. Okay, that's the quick view by which you can understand. I want the hydrophobic amino acids to be represented. And if you don't want the similarity, only the hydrophobic. Now you can easily make out. This is hydrophobic, but serine is not hydrophobic. Okay. Tyrosine, not hydrophobic, but phenylalanine is hydrophobic. So the core hydrophobic residues is labeled here. Then if we want, we can further go into like hydrophobic and aliphatic, right? So then those amino acids are represented. Hydrophobic and aromatic we want. So this we will is going to show why it is coming out to be in the same class. So if we remove off, only the aromatic amino acids are going to be displayed. So based on what you want to explain, right? So if we do not want anything, tiny small amino acids like alanine, serine, proline, then only small amino acids are highlighted here. Charged amino acid like aspartic acid, glutamic acid, histidine, lysine, arginine, those residues are highlighted. So based on the purpose, okay, based on whatever you want to explain, you can pick and then explain. We also have some options here. Show me the transmembrane regions. You can select this transmembrane. Then we will get to know like this is the first helix, 1, 2, 3, 4, 5, 6, and then the 7. So we know that it is having a 7 transmembrane, right? So we have 7 helices. So this is where the first helix is going to start and end. Second helix, third helix. It continues till here, okay? 118, then 119. Okay, this is a block. So all these sequences for the first 60 residues, then from 61 with reference to mouse, it continues like this, then it continues like this, then goes like this, and at the end we have this 352 being present here. Okay, where is the active site present, or is there any disulfide bond formation to occur? Yes, this cysteine and this cysteine, this cysteine and this cysteine. There are 4 cysteine residues, so disulfide, 2 disulfide bond formation possibility is there. Is there any region which can undergo mutagenesis? Okay, so these are the regions where the mutagenesis is possible in the human. Okay, this can undergo a mutation. These regions can undergo a mutation. So that is going to be highlighted. Is there any region which can undergo, again, the natural variants? Okay, so possible mutation, not undergoing with the forcible natural variation that can happen, that it is going to highlight. So if you look into, right, on what basis they are going to say like where this is where the transmembrane is present, because the data is already reviewed. So they know where it starts and where it ends. Accordingly, it becomes very easy to extrapolate or display it over here. Again, where is the helical region? This is where the possible helical regions. So 1, 2, 3, 4, 5, 6, and then the 7 helical regions that are present. Is there a region where glycosylation can happen, right? So this is where it can happen. Is there any beta strand present? So in one of the things, there is this potential formation of the beta strand like this. Yes. So yes, Nandagopal sir.
Sir, by clicking the helix, why the compartments are highlighted? Other organisms,
Other?
only human is highlighted in the helix form.
Yes, because the human is what is the interest. So we are extrapolating from the human to other organisms. From
Yes,
the,
sir.
yeah. But when we say like transmembrane, right, it is going to show for the entire thing. This is where the helical region formation is. So this is one of the ways, or if you want with the cluster W, here also we will get to know like where and all the changes that are going to occur. And in this particular protein DRY, okay, so this is the region which is highly conserved. Entire region from here till here is the highly conserved region. So probably then again we have a highly conserved. Probably this may be a motif. Okay, so when we are doing with the motif analysis or pattern search analysis, then we will get to know like yes, this dry, you can remember DRY. Okay, so this might be a conserved region. Then other regions are not that conserved. There are some gaps being present. There are some mismatches being present like this. So you can take more sequences, okay, based on the interest. That's the most important thing. What made you to take these sequences? That will be the first question asked. Now, if I want to do the next step we will go is, let's say I will go back to the Uniprot, but it is better if I use this Chrome. Now I want to do the multiple sequence alignment, okay, but in my multiple sequence alignment, I want to include both orthologs and paralogs, okay. Then your data set should contain both. Or if you want to do only with orthologs, CCR5 with other organisms. Now I am interested only with other proteins that are there in human, okay, then give it like CCR5 human. But when you are taking, you select the other proteins that are there within the human. So the organism should be only human. Now we have other proteins, CCR5, CXCR4, ARRB2, CCL4, right, CCL4L, other CCR2, right. If you want, we can just take those and then perform the multiple sequence alignment again. We will take only the things wherever we have the CCR, okay, to avoid confusion with others. So CCR5, then CCR2, then CCR3, CCR5, CCR2, CCR3. Do not have like CCR anywhere. Everything else is with the can further reduce with this also. Gene names only it should show. Or can anyone tell me another easiest way by which we can get the paralogs? Within human, other CCR proteins. What is the another easiest method?
Only write CCR human.
Okay, correct. That's one of the ways. Perfect. Only type CCR, and then we have the other that is going to match with the CCR. We have like CCR3 or CCR8. Perfect. CCR9. Any other method when we do not want to go in something like this, and then just collect the closely related paralogs. CCR
and blast P we can do.
Perfect, correct. So go to blast P, okay, and then you provide the human CCR5 here. That is P51681. If when you type something and if it is going to fetch, right, if it shows like job title by itself, then it is clear that it has taken the sequence into account. Okay, so if this description, if it is not going to show, then we need to give it we need to give the entire sequence. Now we select the uniprot Swiss prot here. Okay, then organism we need to say is only Homo sapiens. That means I want to see other sequences that are matching within the Homo sapiens. Then if I want with the closely related one, then I can increase it into blossom 90. Okay, or if you don't want to play with any of these things, keep everything default. That's the safest one. Okay, the people have thought a lot about keeping these default parameters. Okay, so when you are running with the default parameters, even it becomes easy. If I run and if I get the result of whatever I'm expecting, when you're writing a paper, you can say like perform the blast with the default settings or default parameters. If you make changes, then you got to elaborate on the changes that you have made. So if that is not the case, keep the default. So this is the other way. First one, let's deselect everything. First one is CCR5, second one is CCR2, CCR1, CCR4, then CCR8, then CCR9, right? Then CCR6, then this is CCR7, right? So we got all the related chemokines from 1, 2, 3, 4, everything. Then we can select like this and then say like download the first complete sequence. And let's store this one in the file. Right. So we have the all the paralog sequences in the FASTA format. So if you do not know these, okay, when you select, if you do not know all these shortcuts of downloading, right, then it becomes like a very troublesome process. You got to go into each of these files. Okay, if you just follow with the typical procedure, and then you will make it like a FASTA, right? Then you will take this particular sequence, Control C, then you will open the notepad, then paste. The first one is ready. Then you will go back and again you will open the second one in the new pad, right? So this is going to take a huge amount of time. So to avoid this, you can practice a bit, or you can just look into the options that they are providing. So if you spend some more time, because whatever the problem you will be facing, right, so the developers would have thought of and provided some options to save your time. So if you explore, right, then it becomes very easy. If you want to share this file, right, so you can download in the text format or CSV format and then share. Different file formats based on the purpose one can use. Now our paralog sequences are ready, right? So one can have the ortholog sequences also. Now all these are our ortholog sequences because CCR5 that is present in other organisms, right? This is also our ortholog sequences now. We can put all together and run once in the multiple sequence alignment, or we can take all these sequences and then run in the multiple sequence alignment once. So I would give you, like, maybe you can spend another 10 minutes of time with this selection of your sequence and then placing them and generating the results. Okay, this is also a very good tool. Okay, Cluster Omega, we'll use it to get to different formats here. You can explore a bit more. Okay, so don't change the formats as of now with character count as it is, just submit. Then look at how is the difference. Okay, so you assume that you are working on this particular protein. Okay, where is the conserved region in the paralogs? Where is the conserved region in the orthologs? Whether it is matching or not. Is there a region which is matching or not? That you can do a quick analysis and you can let me know. So maybe we will take another maybe a 10 or 15 minutes break now. Okay, so it's now 12:45. So we'll come back at 1 and then we will continue. Okay, because it becomes, session becomes too long. So we'll take 15 minutes break now. Is it fine?
Yes, sir.
Yeah. So you perform this quick experiment. Okay, I want others also to answer. Okay, other participants also to tell me whether which particular amino acid is conserved, both in orthologs and also in paralogs. That is what the question is. Now you know how to take the sequences of the paralogs. Now you know how to take the sequences of the orthologs. Do the ortholog analysis and paralog analysis. Tell me which residue is conserved in both orthologs and paralogs. You can use the same tool. Okay, Cluster Omega itself. Use the same tool. Just if you feel like too many sequences, you take only 3 or 4 sequences, then perform and then say, like, for example, cysteine is conserved even in the paralogs, and cysteine with reference to P51681 as a reference. Okay, so which is there in the 22nd position is conserved even in the paralogs. You can do that one. So we'll come back at 1 o'clock. Okay. We'll continue, sir.
Yes, sir, you can continue. Or wait for another 1 minute.
Yes, sir. Yes, sir. Yes, is there anyone who could to do this? And you can let me know where the conserved residue is.
Sir, 74 to 76.
74 to 76. Okay. So the somewhere around here it will be.
No, sir. LLM and N. 3.
L 3L.
2L and 1 N.
Yes, LLN. Okay, that is this region. I think I have shared the screen. Okay, so for others, the same LLN. So what we can do is zoom it out. So we can go back to this. We can remove everything. Okay, so all these are CCR5, Homo sapiens, CCR2. This is all fine. So this one we will take and copy paste. Right. So these are all over within Homo sapiens different CCRs. So that is paralogs. And these are CCR5 from different organisms. So we can take this, copy, and then paste it. So we have the list of orthologs and also paralogs being present here. Then if we submit this now, rather than making two comparisons manually, right? Even when you do that, so the answer should come something like this. So now the position you mentioned like LLI with reference to P51681, that is this, or this, both are not the same now. So now we can easily make out that this arginine or star, this cysteine is highly conserved in all the organisms. See, the positions may vary, okay? The positions are varying because of the difference in the length, okay? That is why we perform the multiple sequence alignment. Each organism is having a varying length. One is 355, another one is 360, 374, 352, 379, right? 374, so 369, 378. So the length is varying, but we want to see, right, even if the length is varying, is there a place, is there a region which is highly conserved? That is why we perform this multiple sequence alignment. Yes, the cysteine is highly conserved, and whenever we say the conservedness, we need to take some sequence as our reference. So if we take the human CCR5 as our reference sequence, then where is the end of this P51681? That is the 9th one. So then this is 10, 11, 12, 13, 14, 15, 16, 17, 18, 19, 20. So in the 20th position, the cysteine which is there in the 20th position of the human CCR5 is highly conserved in both orthologs and paralogs. Okay, that is how you will make the write-up. And then, as I mentioned, DRY, right? We also have other positions highly conserved, GL, right? Then T, then leucine. So this DRY is also highly conserved in all the things, right? And again, if we take this P51681 as our reference sequence, so this leucine is there in the 128th position. So this is 127, 126, 125. So 125, 126, 127, where we have this aspartic acid, arginine, and tyrosine. These three residues are highly conserved in any of the orthologs or paralogs we are going to take, right? So what is the use of identifying these is we will get to know like the region which is highly conserved, and if there is any mutation, if we observe in any of the protein, if that belongs to like chemokine, right? We have all the possible things here, CCR2, CCR3, CCR5 within human and between the human and other organisms, but we have this DRY conserved. So if by chance, if this region undergoes a mutation, then the entire functionality of the protein will be lost. Okay, the entire functionality of the protein will be lost. So that is where these key residues plays an important role. Now, to bring back the whole thing into an easy perspective or easy understanding of why should we do this multiple sequence alignment, because for many from the different background, right, biology or botany are different. So I usually give this as an example in a way like what is the importance of this conservedness, right? Why should we look for this conserved amino acid? That's the most of the time even the students do ask this particular question, what is the importance? Now to put this whole thing into easy understanding, okay, let us take all these amino acids, whatever we have, okay, we have 352 amino acids in the P51681. So just to make the whole understanding easy for all the faculty or the students, let us take this 352 as 352 faculty members present in your university. Okay. Now, is there any faculty, okay, who is conserved? That means who is there in the university from the beginning until now? Let us say the Department of Botany, whatever. So is there any faculty who is there in the department from the beginning till date? There are so many faculty members who changed. Those you can take it as these gaps or mismatches. There are few faculty who are there for few years and again they will shift maybe to other disciplines, or they are there but not from the beginning. Then you call them as semi-conserved. Let us say the department is there since 30 years. Okay, if a faculty is there since 30 years, then you say like a conserved. Because of that person, the department is surviving. Okay, that's how you can interpret as, because of that single person who is staying since 30 years, he knows the entire strength, weaknesses, or the functionality of the department is happening because of that person. So that is referred as highly conserved. There is a person who joined, okay, maybe 20 years back, but he is not pretty active, but he is just doing his job and helping, kind of a kingmaker. He don't want to be a chairperson, but he helps who has to be a chairperson. There will be that person. Okay, there will be some person in your department. So those are like semi-conservative. That is these two dots. And there may be a person who is there since 10 years, but also working, but not really involved in all the aspects. That is partially conserved. And there are few who will join, who will just come, do their duty, not involved in any of the aspects of the development of the department or the university, just they will come and go. Those are referred as mismatches or not conserved. Okay, so that is how you can interpret this as. Now, whether this conservedness is important now. If somebody asks like, why should we look at the conservedness? Now you have the answer. Because the protein is functional because of these key players. I hope you got the reason why we look for the conserved regions, right? Any doubts?
Yes, sir.
Yes, sir.
The answer that is, these amino acids are very much important for
Yes,
the native or three-dimensional structure of the protein.
Exactly.
That would in turn determine its active site.
Yes,
that is
sir.
its functionality.
Perfect, sir. That is why. So the key faculty who retained in the department since 30 years, he is the responsible guy. That means because of that person, he is there as an active residue or active site region. And because of this highly conserved DRY, as I mentioned, holds other amino acids intact, maintains the structure of the university or maintains the structure of the curriculum and the non-teaching staff or all the aspects of the department they are holding. And then the functionality is happening. Structure is important, then the function is going to happen. So the moment if somebody decides to remove off this key residue, right, then the entire department will collapse. These are the three who are holding. So if we remove one, then the structure is lost. So once the structure is lost, that means the policy is lost. So when your policy itself is lost, the execution is not going to happen. Then the functionality of the entire department is lost. So in the same way, these three residues are there as active residues or highly conserved residues. They are the one who are holding the entire protein, because when we look at the tertiary level, right, next experiments, we can easily make out where they are present and how they are interacting or holding the compactness, right? You can easily make out and extrapolate everything. In your department, you may be a key residue, or there may be someone who is acting like a key residue or the conserved person since 30 years. We cannot say like it is good or bad, okay, because these three residues not undergoing the functionality or not undergoing the mutation, right, the protein is not going to the next step. That's all different perspectives that we can think. If this goes for a change, then the program or the department to go for a next level. Those are all different aspects that we can think of. That is also true. There may be other residues in the beginning which are conserved because they got changed, the evolution has happened. So, but at this point of time, the survival of the function or survival of this protein or the functionality of this protein is happening because of these conserved residues. Okay, so that is how you should look at the proteins. Then it becomes quite interesting. If you look each individual residue as a individual faculty, right, you will get to know like, okay, this guy is, no, from here onwards there is no change, but there is someone who is staying there. Okay, what are they doing, where they are doing, what type of a functionality they are doing, all those things becomes quite interesting. Okay, now we will continue to do this with the phylogenetic analysis. Okay, now how are the orthologs and paralogs are related to each other? So you can put your own sequences. It is not mandatory that you should take only these accession numbers, whatever I have used, but I have given you the formula or the ways by which you can take the orthologs and paralogs. Okay, so as I mentioned in the beginning, you have complete freedom to take, even if you are working on any other protein of your interest, if not with the CCR5, that also you can take. Here, whatever I am doing, right, it is not like I am fixed with these sequences to be selected. It is just going with the flow. Whatever I am getting, I am just picking, because principle analysis remains same, whatever may be the sequence that I am going to take. Now, once we have this, we have an option here to get the phylogenetic tree. Okay, so you just select that. You will get the tree directly here. But as I mentioned, for you to have a clarity in understanding all these regions, right, it is better if we have everything in one particular file. Okay, now this is Homo sapiens. All these organisms are fine. Let's keep all these organisms and store it in another file. Now I have the orthologs ready. Now the paralogs I want. Now the paralogs, I will take this and I will paste it here. Okay, so now Homo sapiens is this. I also have this P51681 repeated, right? So it is the same CCR5 repeated, not to retain duplicates, remove this. Since it is a duplicate, right, you can easily make out here Homo sapiens and P51681 coming out in the same, and the distance it is showing like 0. Okay, so that is why it is a duplicate. Sorry, remove off the duplicates. Now here also retain in a short form so that you can easily make out what it is. CCR2, and this belongs to Homo sapiens. So keep the one word. If you put a gap, only this much it is going to show. Okay, this it is not going to show in the output. Then we have this, make the edit, again keep the CCR1 and retain the Homo. This is another sequence. Then again here we will retain it like CCR4. Since we know homologous, if you want to retain this as Homo, you can, otherwise you can remove it off. That's as long as we can make the interpretation easy, right? You can follow any of your method. That is not a problem. Keep only Homo sapiens. Then again, this is CCR9, right? So retain only Homo. So maybe 4 if we take, that is also fine. Okay, just not a, this is just for your understanding purpose. Okay, when you are working with the real-time project, you can always take some decisions. How many sequences you want? You can take 20, 30, 400, 500. There is no restriction with how many sequences you got to select. Okay, so we have 1, 2, 3, 4, 5, 6, 7, 7 representing the orthologs. Then we have 1, 2, 3, 4, 5, 6. Okay, 6 representing paralogs. So select all, Control A, copy this, and you can go back to the cluster L and go to this input form, then paste your sequence where we have CCR, orthologs and paralogs, everything together. Again, you don't need to make any changes. If you want, we have one option here, how the order should be. Okay, if you select input, human CCR5 remains on the top, that is Homo sapiens, so that the numbering becomes easy. Then you select input order, then say submit. Okay, so the Homo sapiens is there in the beginning, that's the input order. Previously when we submitted, the ordering was different. Okay, so now Homo, whatever the order we have given, still the alignment is made. Then to check, is this DRY shows like conserved. That's one of the ways by which you can validate whether the multiple sequence alignment is done properly or not. Then the conserved residues, everything is fine. Okay, now it becomes very easy to count because the first is the Homo sapiens. So where is this conserved region is, it becomes very easy, 128th. Because when we are working in between, right, so working with this number becomes slightly difficult. If the numbering and reference, if we take from the first row, it becomes very easy. 128, so this is 127, 26, 25. So 125, 126, 127 position of the human CCR5 is highly conserved. And nowhere the human CCR5 is repeated. We have Homo CCR2, 1, 4, 8, 9, 6, 7 representing the paralogs, and these are the orthologs. Same protein, different organisms, same organism, different proteins. Okay, so that needs to be very clear, orthologs and paralogs difference. Then we are interested in looking into the phylogenetic tree. So say phylogenetic tree. Now the interpretation, as given in the morning session, it was mentioned. So always the analysis starts from the cluster. Few will do the analysis from this to this. Okay, smallest cluster are clad to next node, next node, next node, next node, like this. Or few will start from here and then end it up here. Either of the ways gives the same result, same interpretation. Now what we know is whether there are different questions that can be addressed with a phylogenetic analysis. Okay, the method followed is neighbor joining method. Okay, this is a neighbor joining tree without distance corrections, should be fine. We also know the neighbor joining method, how it works. Morning it was mentioned. So it will look for the column with the information and it will generate the tree based on the column information wherever there are some different distances there accordingly. Now it is better to go from right to left. Okay, from right to left in this direction, if we work out, becomes easy, because our intention is not to find the divergence. If you are what you are looking for, that is how the analysis is. If I am looking for the divergence, then start from left to right. If my intention is to look for the conservedness closely related, then start analyzing from right to left. Okay, divergence is left to right, convergence is right to left. Left to right not required now. Now we took the different CCR, CCR1, 2, 4, 6, 7, 8, 9. What we are getting is CCR9 and CCR7 are closely related. Okay, these two are closely related to each other, and the difference between them is don't worry about these numbers. This is the overall normalized length that we will get. Okay, so we will have two types of representing. One is like cladogram, another one is real. If I click real, it becomes like a shrinked one. Okay, shrinked. So again, the values remains the same, but it becomes difficult to interpret. So better always go with the cladogram. Then these two proteins are related, CCR7 and CCR9. Since we are then to this cluster, these two becomes like a one cluster. Okay, to this cluster, CCR6 is closely related. Then to these three, if we make it, if we make these three as one cluster, that is from here it goes, right? CCR8 becomes closely related. Then to CCR8, CCR4 is closely related. Then to this CCR4, CCR1 is closely related, and then the CCR2. Now, overall, if we look into the paralogs, okay, all these are the paralogs. Within human, different protein are the paralogs. Now, if I have to compare only the paralogs, and if I have to include the Homo sapiens also within this, then we can say like the human CCR5 got diverged in the beginning and not formed any paralogous with this. Okay, so the difference is high. That is why it has gone out there. But if we want to do only the paralogous analysis, the Homo sapiens has diverged there because we have included other organisms. Now, how is these paralog sequences related to each other? If that is the question, again you go back, perform the only, that is why I asked you to do only paralog. You can take only the paralog sequences. Now we have all these are our paralog sequences. Take this and put the Homo sapiens also sequence. This is our Homo sapiens sequence. Then paste it. Then you run it. Then you will get to know whether these other organisms, pan, macaca, are influencing the Homo sapiens to get diverged from this, right? So that we will get to know only if we take this. Now what is that we are going to get is, now yes, those organisms are influencing, right? These organisms are influencing this Homo sapiens to show as a diverged from the paralogous sequences, right? Initially itself got diverged. If I take only the paralog sequences and analyze, I will get to know like yes, human CCR5, this is Homo is nothing but human CCR5, is closely related with CCR2. Then this is closely related. Don't worry about where they are present. In the sense CCR1 may be there above this or below this. If the CCR1 comes here, okay, don't say like CCR1 is closely related to CCR2. Okay, and just because CCR1 has come above the Homo, don't say like Homo sapien, CCR5 and CCR1 is closely related. No, it is always, I will just put this one maybe in the PowerPoint so that I can mark, otherwise I'm not able to mark. So
Sir, is there any option to download this tree?
Sir. Yes, sir.
Is there any option to download this tree?
Download the screen submission. We will get the tree data, and this we should put it in again some tools, and that will convert. Otherwise,
We have to take screenshot.
yeah, I have to take this. This is the method. Okay. Okay. Copy. Then paste. It has not taken the. Okay. Now I can mark, so it becomes very easy. Now, what I am mentioning is that is a typical mistake usually observed. That is why I'm just highlighting the things that should not be done. What is happening now? Homo sapiens. This is Homo sapiens CCR5, and this is CCR2. Now the CCR1 is coming over here. If somebody asks like, which is the closely related to Homo sapiens, okay, you always need to check with this clade. Okay, how is the connectivity? And based on that, you should say like human CCR5 and human CCR2 are closely related. Now this CCR6 is there just down below this CCR2, but you should not say like CCR2 and CCR6 are closely related. Okay, so where they are present, it is not important. How they are connected with this branch lengths is what you need to look, not where they are present. They may be present very close to each other, but are they connected with this branch length? That plays an important role. Now coming to this Homo sapien CCR5 and CCR2, the next clause is you go back from this node, what is comes over here. So the next one is over here. Then say like CCR1 is closely related to this clade. Okay, it is not just with the human CCR5. CCR1 is closely related with CCR5 and CCR2. So that's how the interpretation goes. To this clade, it is related. Then this is done. Now we got the CCR1 done. Now go back to this, and then the branching happened, but the other branch has come over here. So that is CCR4 is closely related to the entire clade of this. Okay, so the entire clade of this is closely related. Then again go back. Now the split up has happened, but this is separate. So this is one branching. Then you say like CCR8 is closely related to the entire clade, which includes CCR2, CCR5, CCR1, CCR4, then the CCR8. But coming to the CCR5, whether CCR8 is close to each other? No, it is distinct. In between, we have two other branches coming, that is CCR1 and CCR4. That is how the interpretation. Then if we check with the other things, right? So this is come out over here. Then we can make out like these two are closely related, CCR7 and CCR9. Then to this cluster, CCR6 is joined. CCR6 is joined to this. So that is how the interpretation. Now, if we look into this, if I remove off everything, in the beginning, the divergence has happened. If I have to put this, the other way of interpretation or easy way is I have just rotated this, okay, from the horizontal to vertical now. And now if we analyze in this way, if we take this as a root, right, in the beginning itself, the divergence happened and made it like two branches, just like a tree, right? The stem and then the branching, two branches happened, and the CCR8 got diverged completely, and then these organisms also started. You can actually imagine this as if like all these as the organisms, okay. How you can interpret or feel the phylogenetic tree is you allowed these members, okay, you have labeled these organisms as CCR5, 4, 2, 1, whatever. In the beginning, in the race, you allowed them to go for a run. Then this guy took a left turn, CCR8. Then few of them, these are all the members who started to run in the straight line, and this guy took a right turn. That is nothing but these guys took a right turn. Then after traveling together, then one of the person, that is CCR4, said like I will take a left turn from here. So he took a diversion. Then these members together continued their journey for a while. Then after traveling together, one person said like I will take another left turn, and this guy took a diversion, and these two members continued their journey. Then they decided like, okay, I will take the diversion and you take this diversion. But who stayed together for a long time? That's what the conservedness or closely related refers to. So what we are interested is Homo sapiens. Then you will say like these two guys stayed together for a long period of time in the evolution. When we start the journey here, if we look into, so they stayed together in their journey for a maximum amount of time. So we say like they are closely related. That's how the interpretation is. Now these guys took diversion in the beginning, but they traveled together. Then one guy said like I will take the left turn, but these guys continued their journey. So these are also equally related as these two because the diversion time is the same. So they are closely related. Okay. So in the same way, we can, if we look into this, right, and if we analyze in the same way, they all started the journey, but these two guys said like immediately in the beginning itself, they took a diversion. Then they all stayed together. Now, why should we use or why should we look at all these who stayed together or how long they are together? What is the use of this closely relatedness? All those things. We assume that when they are closely related, right, they should share a common functionality. Okay, because they have a complementarity or they are related to each other, they continued their journey together. It's as simple as two members traveling for a long period of time. We assume that they know each other, okay, or there is a complement. So that is why this phylogeny helps us to know. So most of the functionality of this Homo may be the same with the CCR2 or having a closely relatedness. Percentage of identity between these two may be high. So that is why they are closely related to each other. Then comes the CCR1 like that. Okay, so this is how we interpret. The tree representation may vary slightly with the different algorithms that we are going to use, but the analysis is what is most important. With the technique, slight verification, slight changes may happen, but the way you interpret is not going to change. Now, if we take the animals also, CCR5 from other animals, right? So Homo is different, but to the Homo sapiens, we do not have anything related. Okay, so other organisms like macaca and pepeo, chimpanzee, they are closely monkey and they are related. Then to this cluster, this one is coming out close because of the branching. Okay, we are not saying like though pan is just above the macaca, we cannot say like macaca and pan is closely related. That's what I was referring, because they are not related immediately. If the branching has gone like this from here, then we would have said like closely related, but not in this road, not in this. In the beginning it has changed, so we cannot say like closely related. So then again, it helps in deciding which organism to take, why Homo sapiens are diverse. We can also extrapolate and saying like maybe because of this divergence that has happened in the Homo sapiens, the humans are susceptible for the HIV. Okay, so other organisms are not susceptible because of their divergence, whatever is there, or they are all into one cluster. We got evolved or we got into a different road, so we are susceptible for the disease. These organisms stayed together, so they got the resistance or not susceptible for the HIV. That's how the interpretation goes. So the same application or in the same way one can do the analysis. Okay, taking the coronavirus and with the bats, coronavirus present in other organisms, coronavirus with the human, then you will get to know like what is the difference, how is the evolution happened, what made the Homo sapiens to be susceptible, right? What made other organisms? It is not like in the coronavirus stays only in the humans, right? It can be there in many other organisms. But what made us susceptible? Is it like something the phylogenetic tree generating like this, making us unique or diverged, and all other organisms to be there as one cluster, and they are not getting affected? We got evolved or being susceptible for that, right? So that such kind of analysis can be made from the phylogenetic tree. Any doubts or any queries? I hope you could understand the purpose of doing this, right? Purpose I am always looking for, why we know. Next comes how to interpret. Why is most important than how other stuffs, because you can always take the help of other person for the interpretation. Okay, but if you are a principal investigator, right, and if you know the principle or the concept, then you can always suggest like, okay, if this is what you are doing, so do the phylogenetic analysis, because you know what you can get from the phylogenetic analysis. Then you can always tell to your student or whoever, if you want, you can, this is how I got the plot, how I should interpret. That you, somebody can always help you in the interpretation, but nobody is going to suggest like do the phylogenetic analysis. So that you have to take a decision. That decision you can take only when you know what are the possible experiments available and possible outcomes you will get from that particular experiment. So if you get to know like when we should do the phylogenetic analysis, right, why or when we should do that, if you get that understanding, so purpose of my explanation is solved over there, because you can always get a tutorial or help or collaboration, anything is possible later on to get it done. But if the concept of why we should do, if it is not clear, I can repeat or I can explain it again. Any doubts from anyone related to pairwise or multiple or phylogenetic analysis?
Sorry, there is no possibility to calculate the distances between in the phylogenetic tree, which is created here.
We can.
For this
Yes.
purpose, we have to do another type of analysis, like use to mega or other tools.
Yes. See, we can always, here values are already given. This itself is a distance. Okay. So here if you check these two, 0.005 and 0, that means they are very closely related. Here 0.003 and 0.001. So not much of a difference. That is why this cluster is. This is 0.03. So got into a divergence. How close they are? Okay. So the distance is given already. This is slightly increasing from this, from 0.03 to 0.16, then 0.22, 0.23, then 0.31. This is diverged. Again the clustering happened, so again the values got reduced from 31 to 29, then 28, because these values are closely related. So that means these two should be closely related. 0.31 and 0.31. If you look, right, these are just above these two values. So this has created as one cluster and this has created as another cluster. So the distance is already provided, 0.005 and 0.0085. So that means this is a diverged one. Not having, so initially itself divergence happened, that's what. Later divergence happened. How will you tell? 0.31. That means that much is already traveled, then the divergence happened, 0.31. That
Sir,
is to this point.
CCR8 and CCR6.
Hmm?
These values, CCR8 and CCR6, the
Yes.
values are
Almost the same.
but they are diverged.
True. Because we have in between two other organisms which are closely related, 0.29, 0.28. So you can imagine this as the branch, right? They got diverged as two leaves at the end. Actually, in the phylogenetic tree, these are referred as the leaves.
Okay.
So these are the branches. So leaves are there at the ends, and the branches are the one which are connecting different leaves. So in the same way, we should have put 8 and 6 as closely related if there is no other which is having like 0.29. Then these two would have become like closely related.
These
But we
values are
have,
placed in between them.
yes. So 29, 28 is there between them. So that has become like a one cluster or closely related.
And this is an unrooted.
This is unrooted tree. True. Because to have the rooted tree, we need to have more data. And when it cannot predict the root, so usually it goes for an unrooted tree, but we assume again that there is a root from where the divergence has happened. So this point, maybe if I had to put it back, this. So again, we assume that we can take this as a root and the divergence has happened. And in most of the cases, we will have unrooted tree only, because to say something as a root is not easy. Fixing a root is always too difficult. So unrooted tree becomes easy. We use Mega or other tools, right? The advantage is I can just show you that this is how it looks. So if I open I have just some case studies or projects. I will just check if it is going to accept. Okay, this is the older version. We have option to represent the tree in different ways. So otherwise, not much. Okay, so we have all these statistics. If we say like okay, then the processing is happening and then we obtain the tree. This is with chikungunya data, okay, with one of the collaborator. So we have an option to represent the tree in different fashions in this. So radiant, right, or transitional in the rectangular, transitional in this straight, like a folded one. But your analysis will not make a change. We can just place these things in a different, but the tree representation or the tree understanding won't change in any of the means. Okay, so only the tree representation, circular representation. But let's see, if you look into right, it becomes too difficult to interpret. But goes with what you want to explain or other stuffs. Only the different ways of, right, we can change the positions, we can put, but the analysis remains the same. Okay, whatever may be the method you follow, the analysis will not go for a variation. That's what even in the morning presentation sir has mentioned. There is no strict rule how the presentation should be. We can keep, if somebody wants, like, I don't want these three to be there in the bottom, I want these three to be there on the top. Those things are possible, we can make out. But whether the analysis of the tree is going for a change, no. Once it is fixed that these three are one cluster, these two as one and these three as another one cluster, it remains to be the cluster irrespective of whatever may be your representation. Okay, like CK219296. So even if I represent in a circle, CK219216, right, and CK409 remains to be one cluster only. This will not go for any change. Only the representation is going for a change, but this remains one cluster, this remains another cluster. Whatever may be your representation, that is not going to change at all. Okay, so you can do all these things. Core is how to interpret this. So once it is done, any tool, anything, you can do this. These trees, whatever we have, we are going to generate from the from this. It is we can save it in a high resolution images and other stuffs that we can do always. Can type the captions, other options will be there, but analysis will not go for a change. That's the core. We can also use standalone tools to generate this multiple sequence alignment, and we have like BioEdit and other tools wherein it helps us, just like we did the phylogenetic multiple sequence alignment using this UniProt, where we can say like hydrophobic residues, coloring, all those things. Those are advantageous, I'm not saying like, but when you're doing it the first time, we just want to see which one is closely related. That is our core. I think I have closed that one. Yes, any doubts or anything with this? Even you can generate the phylogenetic tree by taking the nucleotide sequence, okay, and then perform as we when we have downloaded, we also obtained the nucleotide sequence, right? That is the transcript. So this is the one. So you can take this and perform. So for phylogenetic analysis, most of the time people prefer to take the mitochondrial DNA and then perform, okay, because the mutation rate at the mitochondrial DNA is very rare, very less. So one can do the analysis more accurately by taking that, but it is not always the case. Okay, that is just a recommendation. If we are not going to get that, right, if we are doing the analysis of the mutations, then that may not happen. So Whatever the information data is available, we can use that and do the analysis. Any questions or any doubts with whatever we have done so far? I hope you are able to understand the concepts from the beginning, whatever we did, PubMed or BLAST, FASTA, pairwise, multiple phylogeny.
Yes, sir.
Yes, sir.
All right, just want to see with others, because if there is anything you can let me know, right? So we can repeat that, or in each experiment we can go in depth, but that is not my aim is, okay, for each thing. But as I mentioned, my intention is to make you get familiar and just get to know what it is and what we will get. What is the input? What is the output? And what we can expect when we perform this experiment. That is why I'm keeping human CCR5 fixed. And now if you imagine from the beginning, literature, only the abstract papers we obtained. GenBank, we got the nucleotide sequence. UniProt, human CCR5 protein sequence we obtained. We did the BLAST. In BLAST, we have different options to do, get the orthologs or paralogs. First also we did pairwise, taking the human CCR5 constant. Then you can easily make out when I should go for a global alignment, when I should go for a local alignment. Then we did the multiple sequence alignment. There, the strategy again, how to select the orthologs, paralogs, how to look for the conserved region. Then phylogenetic analysis. Orthologs, paralogs, are they related to each other? Are they affecting the tree? Right? All those things. That's the flow from the beginning, whatever we did so far. We'll continue to do. In the next one, we do with the restriction, primer, promoter, pattern, PDB. So we'll do it slowly, no problem.
Sir, I have an thing, sir. Like, can you able to share this recorded sessions, a practical session, so that it will be easy for us to work out also?
Yes.
Because while we are understanding, while you have been saying, I am very pleased, you have given very good examples to understand the concept of bioinformatics. Literally, even though I did my PG in distance, PG bioinformatics, but theoretically we are strong, but you've been giving a very good examples. I have to thank for that, sir. And if you share this recorded session of this practical, it will be easy for
Uma, I think Dr. Uma is speaking.
Yes, sir. Yes, sir.
Yeah, myself organizing secretary.
Yeah, sir.
Yesterday we tried to upload it. By today evening we will upload all those things and we will share you the YouTube link. You can
Sure.
watch whenever you want.
Okay, sir. Sure, sir. Because even today I'm trying for the channel. I've subscribed, but I'm unable to get any of the videos. That's why, sir.
Yes, madam. Yesterday we tried it because of huge data. It is
Yeah,
not
sir.
uploading.
Sure.
Somehow we are doing it. Okay.
Okay, sir.
Once we upload it, it will be convenient to everyone. Okay.
Okay, sir. Thank you, sir. Thanks a lot.
Thank you, madam. Thank you for your feedback. Thank you very much. Yes, any other doubts or anything? Okay, so if there are no doubts, we will stop here. We'll continue.
Sir, excuse me, sir.
Yes, sir.
Sir, can I use this Notepad++ file in Mega software, sir? Can I directly?
Yes. So you
Okay.
put the sequences in the FASTA format, paste them into the Notepad++. Okay, if you want, you can just demonstrate this. So you have the blank one, right? I am using another window here. Okay, that's why I look in that direction. So I am having two screens, and then if I take this and let's say if I go for FASTA, okay, if I want to see here itself, okay, sec temp, whatever. you want only the sequences, right? If you are copy pasting, for example, if I have take 3 and align. So let's say if I have sequences like this, Control A, copy, and then paste the sequences here, then file, save, and then save this particular file in your whatever the name. Okay, let's say CCR file, the dot FASTA. By default, it will be like .txt. But in the notepad, what happens if you type like .FASTA, it will save it as .FASTA.txt.
Okay.
But that is not going to happen in the notepad++. If I just save this here, right, it will be like .FASTA, and you can open this FASTA file in the Mega or any other platforms.
Okay. Okay.
Then you can convert into the different formats. Mega is slightly complicated. Okay, it's not that easy, but yes, it's quite useful. But frankly speaking, in most of my publications, I have not used any commercial tools. Okay, so even I have gone up to like 14 impact factor, but free tools only. Autodoc, free software, multiple sequence alignment, cluster W, pattern, protein-protein interaction, or anything, whatever I have demonstrated so far, all the tools, all the servers, everything is free. Okay, open source. So that should not be a criteria that you should have one commercial tool. So there is no need. You can perform the research entirely with the free softwares or open access, existing online servers like that. It only goes is your design of experiments. That plays an extremely important role. Otherwise, we have free softwares to execute. So in the course also, in these experiments, whatever we have in this internship program, I will use only the tools which are free. Okay, no commercial tools will be used, so that we don't need to worry about how to execute in the future.
Thank you, sir.